To call this, are we okay? Meeting to order. We do have a quorum with 5 members present, calling the meeting to order at 10:01. I note for the record that the following Oversight Committee members are in attendance: Mr. Angelou, Mr. Montgomery, Dr. Rice, Dr. Rosenfeld, and I. We thus have 5 members and a quorum. The chair will entertain a motion to excuse the absences of Mr. Holmes and Ms. Mitchell.
So moved.
Have a motion?
Second.
And a second. All in favor?
Aye. Aye.
Any opposed? Hearing none, the motion carries. Members, the draft minutes from the May 17th Oversight Committee meeting are available in your agenda packet behind tab 1. Are there any corrections to the minutes that circulated? All right, the chair will entertain a motion to approve the minutes of the May 17th Oversight Committee meeting.
So moved.
Have a motion and a second. All in favor, aye. Aye. Any opposed? Hearing none, the motion carries. Our next agenda item is public comment. As we reaffirm at every one of our meetings, public feedback is critically important to our work. We work for the people of Texas, and we appreciate very much when they take the time to come share their experience or their perspectives with us. Do we have any requests for public comment today? It appears that we have none. So now we turn to tab 2. We have invited representatives from 2 CPRIT grantees to provide presentations to the Oversight Committee. Dr. Peter Halton from UT Health Science Center, San Antonio, and Dr. Merchant from Medicine and Therapeutics have joined us today. I'll ask Dr. Wilson to introduce Dr. Halton. Dr. Wilson, good morning.
It is, for me, a personal and great pleasure to welcome and introduce Dr. Peter Halton to the Oversight Committee. Dr. Halton is the director of the Glioblastoma Childhood Cancer Research Institute at UT San Antonio Health Science Center, and was recruited to Texas in 2014, and that recruitment really enhanced the expertise for childhood cancer research in our state, as evidenced by Dr. Halton's more than 3 decades of NIH funding in support of his quest to improve the efficacy and safety of therapeutics for childhood cancer. He's an expert in development of the understanding of the biology and incorporating that into model systems for therapeutic investigation. And indeed, as a CPRIT grantee, heads the Texas Childhood Cancer Xenograph Facility. And I've asked him today to talk about the impact of that facility on childhood cancer research in Texas, as well as to give us a brief update on the Glioblastoma Center. So, Peter, welcome, and thank you for being here.
Thank you, Dr. Halton, for being with us today.
Well, thank you, Mr. Chairman, members of the CPRIT Oversight Committee. We're going to put the slides. Second. As a sort of a slight introduction, today is the 40th anniversary of the reported death of Elvis Presley. And I arrived in Memphis to start work at St. Jude Children's Research Hospital 11 days later. That was 1977. And for the last 20 years at St. Jude, I directed the solid tumor research program. And coming to Texas has been a great opportunity. And as the director of the GRIHI, I have to say that the work that CPRIT does and the funding has been amazingly helpful in recruiting some really star talent to the GRIHI Children's Cancer Research Institute. So we truly appreciate everything that CPRIT does. Today I'm going to give you an update on the CPRIT grant that was awarded to us a year ago to establish a Texas Pediatric PDX course. So a PDX is a patient-derived xenograft, and these are models where we directly implant tumor from a child into an immune-suppressed or immune-deficient mouse. And then and I'll show you the goals of this, are to eventually develop and characterize models that can be used by researchers in Texas and beyond. So the goals of the CPRIT award are to successfully heterograft and propagate between 200 and 250 new patient-derived xenografts of pediatric cancers, and to focus in great part on tumors that arise in Hispanic and underserved children in Texas. As background, childhood cancers are very rare. There's about 12,500 cancers in the United States diagnosed in children under the age of 21, and about 10% of those are in Texas, which is slightly disproportionate to the population size. And particularly in the case of leukemias, there seems to be a disproportionate number of leukemias in patients of Hispanic descent, and the outcome of those patients is somewhat poorer than in other populations. So the idea is to establish these models, to molecularly characterize each model, and provide an annotated de-identified patient database along with the molecular characteristics of these tumors on a public website for researchers to access and to obtain models. And I'll show you some of the progress we've made there. So the idea then is also to provide a training for those individuals who are interested, postdoctoral trainees or graduate students who are interested in developing PDX models, and to learn the process of how to do this correctly, because there's a fair amount of research that's done in this area that really doesn't stand muster. So we feel that a proper training course in how to develop therapeutics is an essential part of it. And finally, to determine whether we can use zebrafish, and I'll come back to this a little bit later, as a primary model for engrafting human cancers, because this would provide not only a relatively inexpensive way of doing this, because an immune-deprived mouse is quite an expensive article, but it also allows us to do very rapid screening of drugs in zebrafish that might be then taken through in the mouse models and ultimately into the clinic. So the application was awarded both to UT Southwestern Medical Center and to the GRIHI in San Antonio. And this basically shows the organization of the current funding. That is, Dr. Steven Peck, who's the Division Chief of Hematology Oncology in the Department of Pediatrics at UT Southwestern, heads the efforts there to consent patients, to make sure that all the regulatory hurdles with the IRB are completed, and then to provide tissue which is sent to us in a very cool box called a nanocool, which keeps the specimen in medium at 4 degrees. It's shipped overnight to us, and we heterotransplant into immune-deficient mice, which are NSGs or NOD SCID gamma mice, which basically have no immune system. And that allows the heterograft to grow quite well. In San Antonio, we have a collaboration between both the University Hospital System and Methodist to collect samples. Methodist has a large transplant program, so that's where we obtain about half of the leukemia samples. And that is headed by Gail Tomlinson, who is the division head of pediatric hematology oncology in San Antonio. The idea is for us in the center to heterograft, to expand these samples and to cryopreserve them so that they can be used for subsequent research both in Texas and beyond. And I have to say that in my career as developing these types of models, we've supplied models to well over 1,000 laboratories throughout the world. And some of the seminal papers that have been reported on the molecular characterization of pediatric cancer have come from these models, as well as development of new therapeutics. So we're involved in expansion, cryopreservation, and storage of these tumors. And then tumors are sent back to UT Southwestern for molecular characterization in terms of exome sequencing and RNA-seq, as well as methylation-seq, to get an idea of both the genomic characteristics as well as the epigenomic characteristics of these tumors. Now, as we subdivide pediatric cancers into smaller and smaller groups, we realize that even homogeneous cancers cancers that we thought were homogeneous can now be subdivided into molecular subgroups that have very different prognostic outcomes. So for example, in medulloblastoma, we now have 4 major subgroups, one of which has a very good prognosis and 2 of which have very bad prognosis. So we need to develop models that accurately reproduce the molecular characteristics if we're going to move forward from cytotoxic type therapy into therapy that is designed specifically for the genetic characteristics of a particular cancer type. And to do that, we have to generate a very large number of models in order to be able to simulate the molecular subgroups that exist within each tumor. So the exome sequencing, the RNA-seq, is going to be done at UT Southwestern, as is the pathological characterization of these tumors. The idea then is to have a website that will make these characteristics available, so anybody who has an interest in a particular subtype of tumor can ask for that, and that will be considered by a committee and distributed to that research group. So in terms of the first year, it's been largely setting up collaborations within San Antonio between different hospitals and getting the appropriate authorization through IRBs, both at UT Southwestern and in San Antonio. We've received 57 samples, 40 solid tumors, 17 leukemias, of which 5 were regarded pathologically as non-viable. 2 were not transplanted due to the paucity of the tumor sample. And we've established 9 new models of solid tumors and 8 models of leukemias. So the quote, take rate in the mice of leukemias is about 50%, about 25% in solid tumors, and I'll come back to that in a couple of minutes. So these are the leukemia models that have been established. They're predominantly from Hispanic patients. 6 out of the 7 patients are Hispanic. We've got 2 samples from 1 patient, 1 at the second relapse, 1 at the third relapse. So there's 8 lines have been established. They're largely B-cell or pre-B-cell, though we have now a T-cell ALL and an acute myelogenous leukemia that are in first passage. We bank germline DNA, so we have a reference to the germline of the patient, so we can determine with some accuracy the mutations that may be involved in generating that cancer. Where I have on the right column, yes, which means we bank germline. P means pending, because at the time the white cell count in the patients was too low to take a blood sample. So when they recover from chemotherapy, then we'll get the germline DNA for doing sequencing from those patients. In terms of the solid tumors, the patient-derived xenografts, 2 have come from Hispanic patients, 2 from African Americans, and you can see the range of tumors that we've got. There's 2 medulloblastomas, one of which is subgroup C, which is a c-Myc amplified medulloblastoma that comes from a very poor prognosis group. Most of these children will succumb within 5 years of diagnosis. And 3 hepatoblastomas, which is interesting because very few hepatoblastoma models from children have been established throughout the world. So this is quite exciting for us to get this opportunity to develop a new model of pediatric cancer that, again, has a relatively poor prognosis in terms of the response to chemotherapy. Then a kidney tumor, a Wilms tumor, which is an anaplastic Wilms tumor. Again, bad prognosis group. Wilms tumor is about 90% curable. The 10% that are not curable tend to be the anaplastic subtype. So again, this is probably the 3rd anaplastic Wilms tumor model that's been established in the world. So we have all 3 of those. Then an ovarian cancer, which is a fairly rare cancer in kids, from a 16-year-old female. Testicular cancer at a primary tumor and a germ cell tumor. So in terms of how we're banking and expanding these models. I'll give you some examples of the sort of cryopreservation. So this is where we freeze down cells in a viable way that they can be put back into mice or put back into cell culture if that's the appropriate vehicle for expanding cells. So on the right column, it shows that from the first passage of a leukemia, 0026, we banked 202 vials. Now, each vial is capable of re-inoculating 10 mice. So in that first freeze down, we have enough to generate about 2,000 mice. And in those 2 or 3 samples, we have about sufficient banked material that could be sent out to upwards of 600 to 1,000 researchers throughout the world. So when these are characterized, we'll see whether they represent very rare subtypes of tumor or whether they constitute more common types of ALLs. But again, can be used for therapeutic use, can be in terms of drug development, therapy development. Then in the solid tumors, we have expanded these. We have basically each vial can reconstitute 6 to 10 mice. So again, we've generated enough material from each of these that in the first passage would be adequate for maybe 60 researchers if they had such interest. So that's one of the objectives, is to expand the models and then characterize them, and then hopefully in the future we will have all the characteristics of these tumors, because that's really one of the primary aims for year 2. So the other thing is that I mentioned earlier that the take rate for solid tumors was somewhat low, 25%, but of 14 out of those 35 viable samples have been implanted within the last 3 months. So the intake of samples is increasing, and these tumors, at least we would imagine about 50% of these will grow. So that will increase our take rates, success rates in terms of generating models to probably close to 40% in the solid tumors. So the goals for next year are to increase the acquisition of tumor samples. One of the challenges, I think, of coming from an environment such as St. Jude, which is a very academic medical environment, is coming into let's say, a less academically accommodating system, where trying to explain what the objectives are and why clinicians should be involved has been somewhat more challenging. The interaction with UT Southwestern obviously is another academic center and has been quite marvelous. What we want to do is increase the acquisition of samples over the next year. So at UT Southwestern, we're going to now incorporate brain tumors and leukemias, which were not provided during the first year. And in part, the brain tumors is because Dr. Larry Kahn, who came from St. Jude, has come to UT Southwestern in Dallas and is going to facilitate the acquisition of the brain tumors. We're going to increase our association with MD Anderson, with Richard Gorlick, who recently became the chair of pediatrics at MD Anderson. Richard and I have worked together for 10 to 15 years, and again, doing work with human tumor xenografts. And with luck, over the next year, we will start to receive samples from MD Anderson to create a much more of a unified Texas PDX system than the one we originally envisaged in the CPRIT grant. We're going to start the molecular characterization of these tumors. With modern-day sequencing, you really have to have 8 samples per lane on a sequencer to make full use of it. So we've now got 16 tumors that we can start doing this, plus 16 germline DNAs. So we're in a position to start exome sequencing, RNA-seq, and pathological characterization of these tumors. We're going to start developing the website to advertise PDX models as one of the goals of the grant. And then I think slightly earlier than we originally proposed, we proposed developing the zebrafish models in years 3 to 5. But with the recruitment of Myron Ignatius to the GRIHI, we've set up a very nice zebrafish facility, both here and Myron, in collaboration with James Armatruda at UT Southwestern, will start to heterograft tumor samples into these zebrafish. So this is a schematic of zebrafish, where you can breed an immune-deficient zebrafish. In these cases, a RAG2 deficient zebrafish. It basically is a bit like an NSG mouse in that it has no real immunity. And the idea is to see if we can grow these tumors better in zebrafish than we can in mice. It'd be a lot cheaper. And to ultimately use this as a screen, you can just put the drugs in the water, and if the tumor goes away and the fish keeps swimming, you're doing pretty well. So it's a nice pre-screen before taking drugs into very expensive models, such as the NSG mice. So one of the aspects of this is you have to have an incubator where you raise the temperature of the fish from 33 degrees, the normal ambient temperature they live in, up to 36 to accommodate the growth of a human cancer. Our body temperature is around 37 centigrade. And at the bottom, you can see just the system where we can, in fact, house about 15,000 zebrafish in a relatively small space. And we can inject them into tumor cells into these fish to see if they grow. And also to study the biology of this, because we can tag different tumor cells with different reporters that tell us where in a cell lineage or differentiation state a cell is, and that tells us whether certain subpopulations metastasize in the fish. So the fish are translucent, so you can image them under a microscope, and they're a really cool model. So why PDX models of childhood, spelled incorrectly, cancer? Well, they maintain the fidelity of the original tumor. If we go for cells, we take a tumor and we put it onto plastic with, in tissue culture, many of the features of those cells are lost. Some of the signaling pathways that are critical to survival in the patient appear to essentially be decreased, and when you put those cells back into animals, those pathways don't come back on. So these tumors faithfully recapitulate the pathology, the genetics, and now the epigenetics. And that's, again, very important, because it's not only what genes you transcribe and the proteins you make, it's how those proteins are modulated later on, and how those proteins modulate gene expression. So epigenetics is becoming a very important aspect of our understanding of pediatric cancers. And what we do know is that cells in culture, their epigenetics change. The methylation state of promoters, which dictates whether a gene is expressed or not, or the histone marks that are associated with specific genes are lost. Whereas we believe that they're maintained to a much higher extent when we heterograft directly into the mouse model. And we have to find out whether the zebrafish recapitulates that as well. So we want to expand the PDX models to simulate clinical heterogeneity more accurately. As I said, what was once a homogeneous disease, such as Glioblastoma, is now 4 diseases. And within each of those 4 diseases, there's probably 3 subtypes that we can try and model to develop more molecularly targeted approaches to therapy. The PDX models are very valuable in understanding the biology of these cancers, and particularly in developing new therapeutics. Because cancer in kids is very rare, and because 70% of children are cured of cancer, the number of patients that go on experimental therapies is becoming increasingly small. So being able to test new drugs in children with cancer becomes more challenging. If we can use the models to identify those new therapeutics that really have a high probability of being effective in specific subtypes of tumors, I think then we can more efficiently use our patient resources that we have to develop new therapeutics. And there are tumors in patients, that present consistent challenges. Obviously, one thinks about glioblastoma in children where the survival is less than 5% at 5 years, or diffuse intrinsic pontine gliomas where the survival at 2 years is less than 3%. But other sarcomas, which when they're diagnosed at an advanced stage or whether they're metastatic, we've made virtually no progress in 30 years. And this shows, in fact, 30 years of clinical trials where we're plotting the probability of survival against time in years. And the IRS-1, the Intergroup Rhabdomyosarcoma Study 1, which was between 1970 and the early 80s, we had about a 30% 5-year survival. And the most recent study, which is IRS-4, we're probably up to 30%, but 70% of those children are diagnosed with metastatic disease when they come into the hospital. 70% of those kids are going to die from that disease. So we still have a long way to go. We have a long way to go in terms of developing effective therapeutics. But the other aspect that's less well understood is that the therapies that we have used, which have been largely cytotoxic therapies and radiation therapy, have left children with very long-term toxicities, chronic toxicities that, as shown on the right column here, about half of those patients who have reached the age of 40 to 50 will have grade 3 or grade 4 chronic toxicities. So in the case of anthracycline use in the 1970s, where these patients probably would have been treated in the 70s and 80s, we didn't realize there was cumulative toxicity. So these patients are at a point where in their 40s and 50s, they have very significant cardiac dysfunction. I think Dr. Greg Alney from my institute spoke to you about his personal history of being a survivor of non-Hodgkin's lymphoma, again, having very serious cardiac complications later in life. So not only do we have to find effective therapies, but we have to find less toxic therapies. And it's our anticipation that by generating these PDX models of pediatric cancers, we can achieve both goals. So there's the group at UT Health in San Antonio, as shown on the left, and the group at UT Southwestern in Dallas is shown on the right. And I have to give great thanks to all of these people. Coordinating, getting specimens, and then heterografting them, expanding them, and characterizing them is a very large undertaking. It's, if we expand to MD Anderson and perhaps to groups in Lubbock, to increase the access to tumor samples, it's going to become a little bit more complicated, but it's a very great team effort. And one of the nice things about team efforts is if we can get UT Southwestern, MD Anderson, our group in San Antonio, and the group in Lubbock to work together, we're covering most of Texas, and I think it'd be very profitable to all of us. So I thank you very much for the opportunity to speak today. Thank you.
Thank you, Dr. Halton. Questions from members of the Oversight Committee? Dr. Rosenfeld? Thank you very much. All right, is your speaker on?
Just a few quick questions. Do you have clinical data that corresponds to your molecular data?
We will have both, yes. So we, at the time we acquire a specimen, we always keep part of that tumor for molecular characterization, plus germline. So we have the original tumor from the patient. We have all the demographics from the patient. We will know what therapy they're on. Obviously, it's de-identified for the purposes of HIPAA, but we will have the history of that patient. We will have the original tumor. We will be able to compare the original tumor with the passage material in either zebrafish or mice to say, are we selecting for subclones within that tumor when we grow the models, or are the tumors that we grow truly representative of the original tumor in children?
But will you have follow-up data after that point? Will you know if those patients lived 5 years, 10 years, 20 years into the future?
I believe we will have that data, yes.
Thank you. Another question is, your take rate was 50%,
With leukemias, yes.
correct? Does that present a selection bias? Because what was different about those 50% that didn't take?
Yeah, so we have the original tumor, and so they will be sequenced as well, because I think that's a really important question. Why do 50% of tumors take and 50 don't? Are they, the tumors that don't take, are they good prognosis patients, or are they the patients that do heterograft, are they the ones who succumb? I can tell you from a retrospective analysis of osteosarcomas, where when I was at St. Jude, we established 10 PDXs from 10 different patients. All of those patients survived, which was counterintuitive. You would think that the tumors that grow in mice would be the bad actors, and that didn't. In the case of osteosarcoma, that didn't appear to be the case.
One more quick question. In the future, when someone requests a tumor, is that a commercial operation? Do you give them free? Do they have to pay to cover costs? Is there a profit? Is this a commercial operation?
Right.
And does it depend whether the requester is a company or an academic site? How does that work?
Yeah, so what we have done, well, let me firstly say that there are companies who do this. Jackson Laboratories develops PDXs, not of pediatric cancer, and sells a mouse for about $1,500.
How much?
$1,500. So if you take our freeze down of leukemic cells, and we charge $1,500, if we charged $150 per mouse, we would generate about $1.3 million just from those 3 samples, if there was that demand. I mean, so I think the way we would look at it would be for academic labs that covering cost would be the approach. But there's progressively in the pharmaceutical industry, there is increasing interest in developing drugs for pediatric cancers. So for example, I have good collaborations with Eli Lilly, Pfizer, and several other companies that have now got subgroups within their cancer development program that are focusing on childhood cancers. Now, if they were to request the models, then I think there would be a chargeback system. We haven't worked out exactly how much that would be.
Thank you.
Dr. Rice?
Thank you very much for the presentation, and again, thank you for being here today. I just have one question, sort of a personal interest. How do you gauge tumor heterogeneity sort of at the time of xenografting and then progressive tumor heterogeneity, and is that something that you focus on?
Right. So there are really 2 questions here. Is, are we representing the clonal heterogeneity of a patient sample when we put it into the mouse? So we sequence the patient sample. So from that, if we do deep sequencing, we can identify subclones within that tumor. So maybe a subclone that has a specific mutation in 1% of the cells that are put into the mouse. So we can ask the question, do subclones become the dominant clone, or do the mice accurately represent the clonal heterogeneity that existed in the patient? So as we go from one mouse, we expand into the next generation and the next generation. We sequence, so we can determine that. We can determine whether the tumor becomes more clonal, whether the heterogeneity remains. Then the other issue is heterogeneity between patients who have the same diagnosis. Again, the same process can be used to say, well, does this tumor represent accurately the genetic heterogeneity of the patient tumor? So in some respects, we're a little bit fortunate in that the genetic complexity of many pediatric cancers is far less than you get in adult cancers. So in my case, I'm interested in sarcomas. And many of those sarcomas are driven by a single event where you get a translocation between 2 chromosomes to generate a fusion gene, which encodes a single protein that basically reprograms the cell. So instead of, as in the case of colon cancer or pancreatic cancer, where you get a series of mutations, where you may have 50 mutations to 1,000 mutations in a cell, the median number of mutations in pediatric cancer cells if you take Ewing sarcoma, is 5 mutations. So 1 fusion and 5 associated mutations, which may have nothing to do with degeneration of the cancer. So they're very different from adult cancers. So in some respects, we're lucky that the clonal heterogeneity may be less in these patients than you would get in an adult patient.
Thank you very much.
Thank you, Dr. Houghton. Any other questions? Dr. Houghton, thank you very much for that presentation. Fascinating, and thank you for your important work. And just like to acknowledge your collaborative efforts across academia and are geographically collaborative, reaching out to Lubbock. And that's something that this CPRIT is very interested in, expanding the collaboration across all of the folks that are joined in fighting against cancer. So
we
thank you for
really appreciate your help
leading the way in that regard. But thanks for taking the time to be with us today. Appreciate it. The chair would like to recognize the presence of Mayor Margot. So let the sounds pretty good, doesn't it, Mayor Margot? That's right. All right. I'd like to call on Mr. Lang, who will introduce Mr. Merchant, another one of our guests who share with us the results of some of the work of CPRIT.
Thank you, Mr. Guerin. It's my pleasure to introduce Dr. Fahar Merchant. He's a 25-year biotech industry veteran and serial entrepreneur. And previously he's founded and led several successful biotech companies, including Protox, Intelligene, Avicena, and KS Biomedics. Over those companies, he's raised hundreds of millions of dollars in investment capital. He's been very, very successful with these enterprises. Dr. Merchant has a PhD in biomedical engineering from Western University. Most recently, he's the founder and CEO of Medicina, which is a Houston-based biotech company focusing on oncology therapeutics. So it's my pleasure to introduce him, and he'll be providing a presentation on the work that's ongoing at Medicina.
Thank you, Mr. Lang. Welcome, Dr. Merchant.
Thank you very much. Thank you for inviting me here today to present to you and talk to you about the Medicina story. Thank you, Michael, for your kind words. And just as an additional background to what Michael just mentioned, was that for the 3 companies that I built and started, each of those companies had a key presence of work that was done here in Texas. So Texas was really the starting point for each of these companies. And I'm really excited to talk to you about Medicina today. And most of all, thanks CPRIT for the incredible hope that you offer through your generosity for these patients who suffer from this terrible disease. So thank you on behalf of the patients and their families as well. So today I'll talk about Medicina. The focus really will be on a disease which is something that you may have heard of recently with the publicity on glioblastoma, which is a tumor that was in the news with Senator John McCain having been diagnosed with this cancer. It's a tough disease. It's by far the most common, most aggressive, uniformly fatal type of brain cancer. And what we're doing at Medicina is something quite unique. And I'll go back to 2 presentations that you've heard of in February by a company called Molecular Templates, where they are developing a very unique, groundbreaking approach to target tumors using very potent toxins. The other company, in May, you heard the story from Inmatics, again, a novel, really exciting field of cancer immunotherapy. We think what we're doing at Medicenna is really combining those two. We're developing targeted toxins where we are able to target not only the tumor, but also the immune system at the same time. So just to let you know, Medicenna being a public company, just a slide here as a disclaimer that I will be making forward-looking statements. But I'll get right down to glioblastoma. And it has been quite a challenge. It's been decades, and there has been very little groundbreaking that has been developed to treat patients with glioblastoma. I'll go back 40 years and talk about these three gentlemen here on this slide, Andy Gesher, Malcolm Stevens, and John Hickman. Each of those were my oncology professors at Aston University, where I was studying pharmacology. And little did I know that these gentlemen were working away on a drug to treat brain cancer. And eventually the drug was commercialized, marketed, became a billion-dollar blockbuster, and the approach used to take this product to the market was the technology came out of academia, and then a funding agency, very much like CPRIT, called Cancer Research UK, essentially funded early clinical development before Big Pharma, Schering-Plough, and then Merck took this product and eventually launched it. So this drug was approved by the FDA in 1999 for glioblastoma. And if you look at it today, despite the fact that it's been over 20 years, or nearly 20 years since the drug was approved, it is now the gold standard. And every patient that is first diagnosed with glioblastoma, they routinely get surgery. And after surgery, they'll get radiation therapy combined with temozolomide. And then after they finish the radiation cycles, they will continue to receive temozolomide for another 6, perhaps another 12 months. But eventually, the tumor always comes back. Patients relapse, and generally, 1 out of 4 of these patients, when they relapse, are good candidates for a second surgery. But by far, the majority of these patients are not good candidates for surgery, and this is where Medicenna is focusing on. Is these patients that have recurred, relapsed, they've failed surgery, radiation, chemotherapy, and the intent is to get the product first approved for these relapsed patients. And then eventually, of course, we'll be looking at other types of brain cancers as well. But let me talk to you about what MD955 is. MD955 is a fusion protein. It's got 2 parts to it. One is a targeting domain that, like a GPS system, will seek out tumor cells. And the other portion is a very potent toxin. So the targeting domain that we use is a molecule called interleukin-4. Interleukin-4 actually is a growth factor. Tumors actually love this stuff because they are able to grow faster in the presence of interleukin-4. So the scientists at the National Cancer Institute and at the FDA, what they did was something really smart. They took this interleukin-4 and attached to it a very potent toxin. And they fused these 2 molecules together to create what we called MD955, in essence creating what we call a very powerful molecular Trojan horse. So if you look at the mechanism as to how the drug works, MD955 actually binds to a biomarker on the surface of cancer cells called the interleukin-4 receptor. So this biomarker is found on cancer cells, particularly very aggressive cancer cells. And we find in the case of brain cancer, for instance, there are thousands of these interleukin-4 receptors that are overexpressed, but you won't find the interleukin-4 receptor in healthy brain. So what we've done essentially is created this molecular Trojan horse where it binds to the interleukin-4 receptor. The cancer cell takes it inside the cytoplasm, and inside the cell, the toxin basically inhibits protein synthesis and kills the tumor. So that is how this drug works. Very well established. There's about 50 papers published on it. So why has it been so difficult to treat patients with glioblastoma? It's been decades since anything meaningful has come out of there. I talked to you about temozolomide, became a blockbuster, $1 billion drug. But all it did was improve median survival from 12 months to 14 and a half months, giving patients an additional 10 weeks of survival. There's a lot more to be done in glioblastoma. So first of all, we find that more than half of glioblastoma patients have tumors that are chemoresistant. And we find with MD955, it's able to target and treat tumors that are resistant to chemotherapy. The second thing about glioblastoma, which is quite unique, is that about 40% of the tumor is made up of what we call the immunosuppressive tumor microenvironment. So what is this? Well, if you look at a stealth bomber, the stealth bomber is an aircraft which is coated with technology so that radar is not able to find the aircraft. Same thing in glioblastoma. It has this tumor microenvironment that surrounds the tumor, sort of a defender of the tumor. So essentially what happens is our immune system cannot see the tumor and attack it. And what has been found recently is that these cells that protect the tumor also express the interleukin-4 receptor. So MD955 is unique because not only will it target the tumor, but it will weaken those defense cells that protect the tumor from our immune system. So that's another big plus. The third thing about glioblastoma is that it's hidden behind a blood-brain barrier, essentially drugs that are taken orally or by IV injections do not effectively cross the blood-brain barrier, and therefore you cannot get therapeutic doses administered in the tumor itself. So to bypass the blood-brain barrier, what we do is actually deliver the drug directly in the tumor. So a tiny burr hole is drilled, very much like what neurosurgeons routinely do is take tumor biopsies in patients with brain cancer. But instead of putting a biopsy needle, they place a catheter that delivers the drug. So MD955 is administered directly in the tumor using a minimally invasive imaging technique. And it's a single administration. So it's a one-time treatment. That's it. So in a sense, what we are doing here is changing the treatment paradigm. We are obviously being a bit disruptive, but I think staying with status quo will likely not do any difference to these patients. So we've got to be a bit aggressive, and that's what we're doing with our approach. And finally, because we deliver the drug directly in the tumor, we are not exposing the tumor to the rest of the body, or rather, we're not exposing the drug to the rest of the body, and therefore minimizing systemic toxicities that is typical in chemotherapy, where you have liver toxicity, nausea, hair loss, and a whole sorts of other systemic issues. So let's look at some data that we have. These are early studies that were conducted in patients that were not good candidates for surgery. And what we found is with one administration of this drug, on the left, you see a tumor, and following a single treatment, the tumor essentially is gone. What we call it a complete response. That means 100% of the tumor has been treated. And what we found is out of the 25 patients, 5 of them had a complete response. And on the right, you see another tumor, again, a single treatment, you see a partial response, meaning the tumor shrunk by at least 50%. So we had a pretty large, about 14 out of 25 patients having a tumor response. And if you look at this plot here, the red line shows you the patients that did not respond, and you can see that the median survival of these patients, remember, these are not newly diagnosed, these are relapsed patients. Some of them had second, third relapses. And you find that these patients had a median survival of about 3 to 4 months, which is typical in this patient population. But those patients that did respond, the median survival was over 1 year. And if you look at the slide, the same figure on the right-hand side, you see data from 52 patients. These were previous clinical trials. But what is quite interesting is that about 20% of these patients went on to live for 3 or even 4 years following one treatment with this drug. So it indicates that somehow the immune system is taking over and attacking the tumor as well, giving these long-term cures in some patients. But then the question is to ask, well, why is it that we had some patients not respond at all? And there are 2 reasons. We found that 1 out of 4 brain cancer patients don't have the IL-4 receptor biomarker, and therefore possibly those patients could not benefit from this drug. The second is that the drug delivery technique that was used in those previous clinical trials was still primitive, in a sense, early stage, and neurosurgeons did not really know whether the drug was being delivered to the tumor or not. It was really essentially a sort of a black box, if you may. So if you look at this slide, the top left-hand slide shows you the tumor treated with the old catheters that were not FDA approved, and you can see a lot of drugs sort of leaking away from the tumor site and not accumulating in the desired region. But now today we have these FDA catheters that prevent backflow. There's FDA-approved software, so neurosurgeons can predict as to essentially exactly where would be the best place to place the catheter, and you can see that you get a much better distribution of the drug. And then as the patient is being treated, what we're doing with our drug is we are co-injecting an imaging agent together with MD955. So now, if you look on the top right-hand corner of this slide, is that as the patient is treated, the neurosurgeon can actually see in real time where the drug is being distributed. So the neurosurgeon can actually adjust the catheter, pull it out, push it back in, and this way we hopefully get much better outcomes. So with this technology in place, with our drug developed, and the protocol in place, approved by the FDA and various IRBs across the country, we are now screening patients in this clinical trial. It's a 43-patient open-label phase 2b clinical trial. So the patients would first be screened for eligibility, and after that, they would undergo an MRI sequence so that the neurosurgeon can plan as to where best to insert the catheter. And then the following day, the patient is treated with this drug. It's a single administration, as I mentioned, and then after that, the patient essentially is followed up for safety, for survival, for tumor response, meaning tumor shrinkage, and so on and so forth. And I'm happy to say that as part of this clinical trial, the first patient that was enrolled in this study was a Texan in San Antonio. So in addition to 2 sites, 2 hospitals in Texas, one in San Antonio and the other in Dallas, Southwestern, we're working together with other top-notch hospitals across the country, 2 in California, we have 2 in the state of Ohio, and then New York, North Carolina, and Florida. So these are world-class centers, and really proud to say that this technology has been really well received and had the opportunity to work with key opinion leaders with this drug. So this is really just talking to you about our brain tumor program. But then if we look at data that we've collected from I would say about 2,000 patient biopsies. These are tumors outside the brain. What we find is that there are 20 cancers that overexpress the interleukin-4 receptor. And as the tumors get much more aggressive, the level of the interleukin-4 receptor increases dramatically. So these are examples of 8 different cancers that I put out there. And what we're doing now is developing a next generation version of a drug of MDNA55. We call that drug MDNA57. It's different. It's got a much more specific targeting domain, which is a new version of interleukin-4 that we've in-licensed from Stanford University. And we have a new payload, which is a fully human payload. It's not from bacterial sources that can be immunogenic. And therefore, this payload, we've licensed that from Hebrew University of Jerusalem. And combining these two technologies, we are now working together with Dr. Michael Rosenblum at MD Anderson to develop this new drug, which I think is really exciting. So we have, as I said, MDNA55 is our lead program. It has multiple opportunities. As you can see, the first indication we're pursuing is recurrent glioblastoma. And then the plan is to commence a study in brain metastases. Now, we've heard before the issue with patients that get treated that might have breast cancer or lung cancer. What actually eventually these patients succumb to is when the tumor spreads to the brain. And we found that, again, more than half of the patients that have tumors that spread to the brain also have the interleukin-4 receptor. So that's another huge opportunity. And then, of course, we're looking at newly diagnosed glioblastoma as well. But I think the real challenge right now is pediatric brain tumors. And if you look at pediatric tumors, today, brain cancer now in children is the leading cause of death due to cancer. And these are challenging, especially tumors such as diffuse intrinsic pontine gliomas. When we took patients' autopsies from 12 patients and analyzed them, we found that all of these tumors from these children express the interleukin-4 receptor. So we are looking forward to doing some work in that space as well. So we have, with the CPRIT funding, made substantial progress. First of all, with the $14 million CPRIT grant, we were able to secure matching funds of $11 million. So this basically fully funds our program. We've put ourselves here in Texas with a base with 2 C-level executives now operating and managing the projects now here in Texas. We have also manufactured MDNA55 here in Texas, and I'll talk to you a bit more about that later on. And as I said, enrolling patients in the clinical trial at 9 centers. 2 of those sites are in Texas. And of course, our first patient was treated here in Texas. As per our last financial report, the company announced that we have enrolled 10 patients to date out of the 43. And indeed, we've got some really exciting milestones coming up over the next 12 to 18 months. We are expecting to complete enrollment in this clinical trial in the next 6 months, and thereafter come up with interim top-line results from this clinical trial. And then if the data is positive, go to the FDA for an end of phase 2 meeting. We are also planning to start additional clinical trials with MDNA55 in metastatic and newly diagnosed brain cancer, and later on pediatric brain cancer as well. And of course, with our collaboration with MD Anderson, demonstrate proof of concept with this new drug called MDNA57. And with this level of activity, and that keeps on expanding, and therefore concurrently we'll be expanding our footprint, both development and commercial footprint in Texas as well. So I'll talk to you about manufacturing first. This particular project started before the CPRIT grant was awarded. We established the manufacturing process at the College Station facility, which is now currently owned by Fujifilm. And what we were able to do was improve the manufacturing process yield to the state where we now have a fully commercial-scale manufacturing process established for MDNA55. To give you context, in fact, only a small 50-liter fermenter that's all we need to make our drug. What it does, it makes about 3 grams of the drug, and you'd say, well, 3 grams is pretty small amount. But what is unique is this drug is potent and targeted. We need only 1 gram of this drug to treat 10,000 patients. Okay, so you can see that as far as the cost of goods are concerned, that's really insignificant. So in addition to our manufacturing alliance with Fujifilm, we work with clinical CROs, both of them based in Houston. One is MedSource, the other is PharmaLam. With respect to all the images for patient enrollment and follow-up of their tumor size to see if they're responding or not, we work with a company in San Antonio called Intrinsic Imaging. And then, of course, we have a collaboration with Dr. Rosenblum at MD Anderson, and the clinical programs that we have currently enrolling patients at UT Health in San Antonio and UT Southwestern in Dallas, working there with Dr. Brenner and Dr. Patel. So if you look at the kind of impact CPRIT makes to this particular project, where are we really? As I said, we are developing MDNA55 and MDNA57. Both of them are targeting one receptor, the interleukin-4 receptor. This particular biomarker is overexpressed in 20 different cancers, affecting 1 million cancer patients every year. So we have received a generous $14 million grant from CPRIT, matched it with another $11 million that we have raised. And with this drug, MDNA55, we have the potential to treat 250,000 patients with either glioblastoma, metastatic brain cancers, or pediatric tumors. As I said earlier, we now have a manufacturing process where 1 gram of this drug will treat 10,000 patients. And the potential upside market opportunity for this drug, MDNA55, for different kinds of brain cancer is $2 billion. And I think the biggest number of them all is infinity. The infinite hope that CPRIT provides these patients and their families from this terrible disease. And for that, thank you for your support and help. And I'll take questions.
Thank you, Dr. Merchant. Have any questions from our Oversight Committee? Dr. Rosenfeld?
Thank you. Enjoyed your presentation.
Thank you.
A couple quick questions on the scans you showed of the brain of the responding tumors.
Yes.
Brought up the question whether only peripheral brain tumors are eligible. Are tumors that are more centrally located, can they also be treated?
Yes, as long as a catheter can be placed safely, that patient would be eligible for study. Sometimes even superficial tumors that are very close to the cortical surface, that have infiltrated the cortical surface, those, although they're easily accessible, those patients sometimes could have leakage into other areas and therefore not be ideal. And similarly, you might have a tumor that's very close to the ventricles, in which case the drug could potentially leak out into the ventricles. So there are certain types of tumors that are, if they're too close to the ventricles or are not accessible by a catheter placement, then those would not be eligible.
Have you been able to treat any brainstem tumors?
Not yet.
Okay. And I was wondering, in your presentation, you said several times that only one treatment is needed. You emphasized
Yes.
that.
Well,
And-
yeah, go ahead.
And I was wondering why only one treatment is, because, you know, when you think in treatment of tumors with monoclonal antibodies, you would say, boy, would I ever give one round of Rituxan?
Right.
And I'm just wondering, when you look at your response rate, would there be a rationale for repeated dosing, and would that fit better into a commercial plan?
Absolutely, really good question, because, truly, you're completely right. The protocol being the study that it is, obviously the feedback we receive from neurosurgeons is, well, Fahar, the patient had a partial response, the tumor is coming back, can't we treat the patient again? Unfortunately, the protocol is very fixed into a single treatment. We have to standardize a few things, but we understand that at a commercial scale, there is no reason as to why repeat administration would be prohibited, unless, of course, the patient simply didn't respond to treatment. But if the patient responded, there is a good chance that the patient would respond again when the tumor came back. Absolutely.
All right. Any other questions from the Oversight Committee? Dr. Merchant, thank you very much for your life-saving work and for coming and sharing your story with us today. Appreciate
Okay,
it.
thank you very much.
The chair now recognizes Mr. Roberts to provide the Chief Executive Officer's report. Mr. Roberts.
Thank you, Mr. Guerin. First, let me start off by thanking our friends at the Texas Higher Education Coordinating Board, Commissioner Paredes in particular, for allowing us the use of these great facilities while we have to accommodate the special session of the legislature, which is, of course, gone, but we didn't know that. I'd also like to call to your attention a document that was placed at each of your chairs, a notable CPRIT-supported research and prevention accomplishments. This is a compilation of the summaries that you now receive in those monthly reports that we provide you in months that you do not meet. This is getting to be quite a fun document. I think that part of the report, if you only focus on one thing in those monthly reports, I encourage you to focus on those because it is absolutely amazing the quantity of work, neat work being done by the grantees. And since we started that report, this is a summary of it, so you can expect to see this on a recurring basis. The next item that I'd like to talk to you about briefly is where we stand with respect to personnel. Right now, all of our current 32 positions are filled with permanent or contracted personnel. The 3 contracted positions we expect to be filled with permanent employees on September the 1st, which is the first day that we can officially hire individuals after the governor's moratorium. The 3 new compliance positions that were authorized by the legislative session are posted, and we hopefully can fill them by the end of September. Immediately behind my memo is the familiar chart on 2017 grant award funds available. And I'd like to call your attention to the lower right-hand corner. If you approve the recommendations from the peer review process and the Program Integration Committee, we will be leaving $2.6 million on the table. Unfortunately, we had a recruit decline an award for $2 million. Otherwise, that margin would be right at $636 million. I've got to tell you that as the administrator of the agency, that's cutting it a little bit closer than I would like. So in future years, my recommendation that I would like to implement is to plan on having a 1 to a 1.5% reserve buffer to make sure we don't go into the red. In terms of dollars, that would range from 2.8 to about $4 million. Clearly, there's some flexibility there. Any of that money in the reserve, if we end up not using it in any given year, we do have the authority to roll it over to the next year, and I feel fairly confident that by the time we sunset in 2023, we will have exhausted all of those unexpended balances, so we won't be shorting any of the programs with that. The next item on my report on page 3-5 in your books is a short memo to inform the Oversight Committee of an activity that is going on, and it is to review or to scope out the possibility of a complementary RFA program that I suspect would be related to the scientific research program. And because this is a new initiative, a new approach, I have requested that Mr. Guerin and Dr. Rice participate in the scoping process. We have had one meeting related to this. One of the stimulants for this, you might recall, is Ms. Susan Dawson spoke to us, I believe, in February about some ideas she had for a new proposal, funding mechanism. This is also something that I and some of the staff have been interested in doing, not necessarily this, but a new approach to complement what we're doing. Again, I just want to inform you folks of this activity, and you can anticipate hearing more about this in the months ahead. Dr. Rice, Mr. Guerin, I don't know if there's anything I'd like to add to that.
No, I would like to acknowledge that Ms. Dawson is here today, and appreciate very much her input and how she's helped us think through this initiative. Thank you. Ms. Dawson.
The next item in my report on page 3-7, CPRIT funding scenarios through FY 2023. As you know, the legislature authorized us to have the extra 2 years that we were requesting. So that gives us some certainty in the funding availability in our out years. Ms. McConnell, our Chief Operating Officer, and I discussed this with each of the subcommittee meetings leading up to this. We would be happy to go into greater detail, but unless there are any questions or desire to expand, I won't repeat what has already been presented, other than to say that this document, these projections can be used to inform the Oversight Committee in the program priorities discussion that is going to be coming up and will be talked about again in a little while, and is also a document that you will see on a continuing basis as we go forward. If there's no comments on that, the remainder of my report, you have the most recent dashboard update provided by Ms. McConnell, along with the 2 CPRIT activity reports from June and July during the time since you met last. With that, Mr. Guerin, I'm available for any questions, comments, or suggestions, and completes my report.
Thank you, Mr. Roberts. Any questions? I just would like to, as we look at how unpredictable the legislative process can be, and how, reflecting back on this past spring and the work that Wayne, you and your entire team did, keeping the legislature fully informed and working with them through the legislative process to put us in the very good position we are in today. Just want to acknowledge that again and thank you all. It's harder than it looks from the outside, and you all did an outstanding job. And we look at this funding schedule through 2023. We look at our ability to plan going forward, removing a lot of the uncertainty that had hung over CPRIT going into this legislative session, all resolved very much in favor. And I believe it's our legislature now understands the important contribution CPRIT's making, and you and your whole team did an outstanding job of educating them, responding to their queries, and putting us where we are. So,
well, we appreciate that. With that, I've got to tell you that, frankly, it was a pleasure during this session as it progressed. I'm not sure every state agency can say that, but we have really outstanding legislative champions, and Senator Nelson on the Senate side, Representative Sarah Davis on the House side, along with Representative Zerwas and Senator Kirk Watson. They're the ones that made this, I don't want to say an easy session, but a very pleasurable session.
All right. Thank you, Mr. Roberts. Appreciate it. And if no further questions or comments for Mr. Roberts, I'd now like to call on our Chief Compliance Officer, Mr. Burgess, our own Inspector Yover, that diligent, determined, keeps us on the straight and narrow, as well as our grantees. Does a great job for us.
Thank you, sir. You'll find my compliance program update behind tab 4 in your meeting booklet. I realize we have a lengthy agenda today, so I'll be brief. My report highlights many of our compliance activities and initiatives, our delinquent reporting, our grant monitoring with our desk and on-site reviews, audit tracking, grantee trainings, as well as our FY18 grantee risk assessment implementation. And we reviewed that process with the audit subcommittee a week or two ago. And so I can answer any questions you may have about that report that was made available to you. If you could flip over to 4-4, direct your attention to that graph. This is a graph that I've provided for you the last few meetings, and this shows the last 13 months, July to July, kind of our self-imposed 5% non-compliance threshold, and we have, as you can see, come under our target. Hit it close a little bit in May and July, but we were indeed under the 28, I think it's 28.5. And if you flip over one more page on 4-5, something Mr. Guerin said last meeting in May, just really wanting to put in perspective when we talk about delinquent reports and there being 20 or 30 or 50, giving some historical context to that. And so I provided this chart, and it shows the FY 15, 16, and 17, and this is a monthly average. So in FY 15, we were averaging about 78 delinquent reports per month. In FY 16, that was reduced by about 73% to 21 a month. And then FY 17, and that is through July, the figures, and we reduced it another 43% to average of 12 per month. I did also want to note that we don't have complete data for FY 14, but I was able to find the last month in FY 14, August of 14, we had 483 delinquent reports. So you can see we are on the right track with that. But that is a testament to the hard work of our compliance specialists, our grant accountants, our program managers. It's definitely been a team effort, and great working with them.
That's extraordinary work, and
Any questions for me?
major progress since
FY
you took over
14.
that responsibility.
And that concludes my report. Any questions for me?
All right. Any questions for Mr. Burgess? Thank you. Thanks for your great work. And I'd now like to recognize Dr. Wilson to provide the Chief Scientific Officer's report and introduce the Program Integration Committee's grant award recommendations. And I direct you to tab 5. Welcome, Dr. Wilson.
Thank you. This report will come in two components. The first is the grant recommendations for this quarter based on the PIC committee's recommendations to you. And then the second will be a review of our current and proposed RFAs as requested by the Oversight Committee. The PIC committee has recommended 52 awards. We're bringing 51 to your attention because one of those awards, as I'll mention during the presentation, was withdrawn. One of the recruitment awards was withdrawn between the PIC and this meeting. So 51 awards, $79 million and $481,933, 6 slates. And as before, we'll go through each of these slates, if this is agreeable, stopping with each slate presentation for questions, and then a final review at the end. So on page 10 and 11, the first slate is an early translational research awards applications. These were reviewed in the first cycle of this year, and 3 were held because of uncertainty about our ability to fund these fully. It turns out, quite fortunately, that that is, CPRIT is able to fund these with a slight budget modification or reduction of 8.5% of the recommended funds. These 3 are presented again on page 11, and 2 of the early translational research awards are addressing targeted priorities. Yes, Craig. Okay.
In the main, yeah.
Right, so Dr. Rosenfeld's referring to the, I think the scientific officer's memo in your earlier information about recruitments. And Mission Links, early in the history of CPRIT, was an attempt to, was a RFA for recruitment of individuals who would solve a particular need in an institution, but may not have the academic track record to qualify for an established investigator award. These, for reasons that my predecessors and you deliberated on, have not been continued. Sorry, back now to the grants recommendation. Are we okay? Okay. So we discussed the early translational research awards that were held over from cycle 1. And now I'm moving to a group of 8 individual investigator-initiated research awards. Again, these were reviewed in the first cycle of this fiscal year and recommended by Scientific Review Council, but we held bringing them to the Oversight, to pick in the Oversight Committee until we were sure there were sufficient funds. There has been an 8.5% reduction in the funds that were recommended by the Scientific Review Council. Among these 8, 4 address targeted priorities and are across a variety of important cancer tumors in terms of where the discovery is focused. Moving on to the third slate, you'll recall that every year CPRIT has had an RFA calling for high-impact, high-risk research awards. These are $200,000 awards over 2 years to stimulate new ideas for cancer important research that may not have an extended track record of preliminary data to qualify for an investigator-initiated award. It's notable that these awards have attracted investigators who are not traditionally associated with cancer research, and also institutions who are not prominently award recipients. And to that point, there were 19 high-impact, high-research awards recommended by the Scientific Review Council out of 143 that were applied, a 13% success rate. 8 of these 19 addressed direct targeted priorities. And again, as in the past, while large cancer research institutions are represented, it's notable that Texas A&M, Health Science Center, A&M Campus, Texas Tech, UT Arlington, UT Dallas, University of Texas San Antonio, main campus, Rice are all represented here, again, to the point that this extends the opportunities to get into cancer research across the landscape of Texas. The second RFA reviewed in this past cycle were for core facility support awards, and these appear on page 14, 15, 16, 17. There were 24 applications for core facility support awards. You heard today from Dr. Halton about the childhood xenograft core facility, which was awarded last year by CPRIT. 7 of these applications were recommended by the Scientific Review Council, and I'm going to highlight 2 and then open up if you have questions about the others. The first I wish to highlight is on page 15, the cyclotron radiochemistry core facility for pediatric oncology that's presented to, or from UT Southwestern, 170638. This core facility builds on a previous CPRIT infrastructure award to build a cyclotron. A cyclotron is required for making the radionucleotide and radiopharmaceuticals that are used in PET imaging. PET imaging is a very important cancer tool for evaluating the function of a cancer, in contrast to CT scans and MR scans, which give you the anatomy. And so in the context of childhood cancer, this is a particularly important investigative tool for both understanding the response to therapeutics as well as the development of new therapeutics. What this core facility will provide is the ability to use the cyclotron to develop the pharmaceuticals to do these investigative clinical trials. And it's important to note that the proximity of the cyclotron to the actual imaging center is quite important, and that's what's been developed here. The second core facility of this group of 7 that I wish to highlight is on page 17. It's the Data Science and Informatics Core for Cancer Research from University of Texas Health Science Center at Houston. This is a particularly interesting core facility that builds on a established area of commitment by the UT Health Science Center in bioinformatics, and a quite extraordinary facility that is now proposed to support big data analysis for cancer. And I think as all of us recognize that as the genomic era and now proteomic era has come, as well as the importance of having clinical data to correlate with these, we're in the process of being able to amass large amounts of data. And the challenge is how you take these large data sets and gain knowledge. And so that's what this facility is all about. It provides the hardware for accommodating the large data. It will have 1.5 petabytes available. Now, I didn't know what a petabyte was, but it turns out that it's 1 million gigabytes, and I think all of us know what a gigabyte is because we can carry it around in our pocket, but think now of 1 million of those now available for big data research. And then it will also provide access to the computational scientists to support the interface between cancer investigators and these data sets. And importantly, an outreach for education to investigators on how to actually use computational biology. As I mentioned, there are 7 recommendations in this slate. I'll draw to your attention that 3 of the 7 have had budget reductions based on recommendations from the Scientific Review Council that have been carried forward by PIC. These reflect opinions by the Review Council in terms of using these funds optimally. One in particular is notable, and that's on page 17, the Individualized Pediatric Tumor Analysis Center of Texas. The Scientific Review Council recommended that rather than a full core facility award as requested, a pilot funding of $200,000 to allow this group to pursue the development of what the review panel felt was a very important opportunity in Texas, but not one that the investigators were fully ready for complete operation. And the hope was that this pilot funds would allow this potentially very important facility to come back in a future date for a more competitive application. I'll stop there for a moment. Yes, Dr.
One
Rosenfeld.
quick question on a individual investigator research award.
Yes.
I noticed there was an $800,000 grant for Kenneth Westover at UT Southwestern
Yes.
on looking at KRAS G12C. And it just the reason it caught my attention is he's published a series of papers on this, including one last week. And I was wondering what's different in this new funding that wasn't in already published, because he's pretty far along and has SAR on his small molecule inhibitors and crystallization of the target.
Dr. Rosenfeld, you're way ahead of me in terms of your review of the literature, so I cannot comment on Ken's most recent work. I will say, as you know, he's a CPRIT scholar, and pursuing a line of investigation in this area of targeting KRAS mutation in lung cancers, which, as you recognize, is a key challenge in oncology. I cannot speak to whether he's completed the work. Obviously, we will review that as his contract goes forward, if you approve. But at the time of the review, his work was considered to be at a cutting edge and very meritorious, as you know. And it's great that he's actually making the progress that we all hope for. Well, as I say, you're much more up to date on his publications, and we'll take a look at that and have to get back to you because I cannot speak to that. Yeah. Other questions? Because we're now to the seventh slate, I think. Oh, sixth, I'm sorry. Apologize. This is the fifth for established investigator recruitment awards. The PIC has recommended 2 established investigator awards. These are really quite remarkable individuals that I'm telling you about. The first is Christopher Amos on page 18. Dr. Amos is being proposed for recruitment to Baylor College of Medicine. He's currently at Dartmouth. He is recognized as an international expert in the use of quantitative sciences for genetic analysis. Again, big data evaluation based on large data sets that are now available through population studies that he has catalyzed across actually the world. And the second is Dr. Carlos Arteaga, a physician scientist who's being recruited to UT Southwestern from Vanderbilt Medical Center. Dr. Arteaga is the incumbent director of the UT Southwestern Simmons Comprehensive Cancer Center. And he is a breast cancer investigator, physician scientist who's recognized across the world for his research focused on hormonal responsive malignancies, and is not only doing laboratory-based research, but also developing clinical trials to test these discoveries in patients. And finally, but certainly among the most exciting of these slates are the first-time tenure-track investigators who have been recommended. There are 12 of these individuals, one from Rice, one from University of Houston, one from Texas at Austin, Health Science Center Houston, Health Science Center San Antonio, and 7 from UT Southwestern. I will allow you to peruse the specific descriptions of these individuals and point out that 5 of these 12 address computational biology priorities and 2 childhood cancer priorities. And I mentioned earlier that 170064 on page 21, unfortunately or regrettably, chose to decline the CPRIT award and is going outside of Texas. Mr. Guerin, that's those are the 6 sites that that we're bringing forward from the PIC. So the bar chart on page, I believe it'll be page 26. At the top, you'll see the academic research program priorities listed along the top. And what you see are the number of awards that address these specific priorities among this slate of 51.
Mm-hmm. So the bar, the height of the bar represents the number of awards, not the dollar amount of the awards.
That's correct, yes.
Mm-hmm.
The dollar amount is actually provided, but the actual height of the bar is the number of awards.
Mm-hmm. Could you just talk for a minute about the challenge in getting more awards in the cancers of importance in Texas and in disparities columns?
Well, the cancers of importance in Texas are focused on, in the past year, lung, liver, and cervix cancer, and again, are prominently represented in the CPRIT portfolio. I don't think that there's actually a limited number or breadth of coverage of the CPRIT awards for these particular areas, and would opine that actually the recognition of the research community of the importance of these diseases has led to, for example, last year, 3 of the Myra Awards were focused on lung cancer, and then the year before, a Myra Award on hepatocellular cancer. So I guess my response is that I think that the portfolio is well represented.
Mm-hmm. And the period of time represented by this chart is an anomaly.
Well, I wouldn't express it as an anomaly, only in that there are a number of awards here, 5, but this represents just the past 6 months, mm-hmm. the second cycle of the CPRIT awards.
And the disparities, you'd say the same thing about?
Well, the disparities is a matter of I think similar opportunity that is not yet as fully covered necessarily by the CPRIT portfolio, and one that I think is recognized as one that continued to think carefully about. One of the things that that we've been particularly interested in is accelerating the accrual to clinical trials as it relates to traditionally underrepresented populations, and as I'll mention in our next presentation, we have some initiatives to try to stimulate that, mm-hmm. with a call for specific strategies to increase accrual in those populations.
Mm-hmm. Could you speak to the prevention and early detection as well?
Yes. This is an interesting problem because Texas has some very strong prevention research programs throughout the state, but we don't see the same number of applications as we do from traditional biology. In an effort to stimulate those, Dr. Kripke initiated a targeted RFA, which has been continued, and in addition constituted a targeted panel to review these, and it's led by Dr. Tom Sellers, a world's expert in population science research. So I think that this is an area where we continue to, haven't completely solved the opportunities that still are out there for generating applications in this area. One of the things that we've done in collaboration with Dr. Garcia is to encourage the communities that are addressing prevention service delivery to begin to think about ways to interact with the academic researchers to take advantage of those initiatives as a laboratory, for example. And so our most recent RFA that's being reviewed now has a particular call for CPRIT's interest in taking advantage of those population demonstration projects that are supported by Dr. Garcia's program.
Mm-hmm. Thank you. I appreciate that. Any other questions for Dr. Wilson? Mr. Angeli?
Dr. Wilson, is there any way to quantify any residual benefit that these highly qualified individuals bring to the state, like NIH grants or corporate grants or graduate students that may move with them to Texas?
Yeah. Yes, there is, and one parameter would be peer review funding from outside of Texas. We looked recently at the 135 CPRIT scholars who have come to Texas, and to date, they, as an aggregate, have brought peer review funding that exceeds $225 million. Remember, too, that a large portion of those are the early stage investigators, and so this is actually, I think, one of the parameters that we will continue to follow. The other is the actual reputation that these individuals bring to the state, and you're obviously aware of some of the more prominent individuals that have been recruited. Just the folks that I mentioned today, Dr. Arteaga. Dr. Arteaga, who was coming to UT Southwestern, will bring to the state a halo of recognition for his work in breast cancer. He is the leader of the, co-leader of the annual breast cancer conference that's held in San Antonio and considered across the world as the go-to conference for breast cancer. So again, two things: one, tangible numbers, and second, these less tangible but very important halo effect on what's happening in Texas.
So in order to get that data, first they have to move to Texas and then discover the residual value, or is it possible that we can identify what grants these individuals may already have, wherever they may be?
Yes. So we know that as part of their application, both Dr. Arteaga, for example, holds a, and has for the past, I think, 15 years, a specialized program of research excellence from the National Cancer Institute in breast cancer. This is a $2 million a year NIH award that focuses on breast cancer research that he holds. Dr. Amos is equally well, in fact, actually significantly well-funded for international collaborative studies in population science. And so those, that information is actually part of the review process. Okay. Yeah.
Thank you.
Obviously, what we're really interested in is when they get here that they continue to build that. And what's been exciting is to demonstrate that even though these are very generous awards, these investigators are continuing to apply those resources to build bigger programs.
Very exciting to ponder the contribution to the human capital in Texas that this slate represents. All right, if there are no other questions for Dr. Wilson, I'd like to call on Mr. Burgess, our Chief Compliance Officer, who will provide the compliance certification report for all of the proposed awards at this time, including the product development and prevention awards in advance.
Thank you, Mr. Guerin. Please see page 123 of the proposed grant award booklet, memo dated August the 2nd for my full compliance certification report and details. And my report was also made available in the portal. As part of the compliance certification, we review supporting documentation for each peer review and review council meeting. The supporting documentation includes the compliance pedigree, and that outlines, details each step in the application and award process. And depending on the mechanism, it could be anywhere between 40 and 60 unique steps in the process that we document and certify. We also review the third-party observer reports, the sign-out sheets, the conflict of interest sign-out sheets, attendance sheets, and post-review statements. There are lots of moving parts in the compliance certification, and this was quite a large number of awards this time around. I also attended the Program Integration Committee meeting on August the 1st. As an observer, and I confirmed that the PIC review process complied with CPRIT's administrative rules and statutes. Having reviewed the supporting documentation for the 6 academic research award mechanisms, the 1 product development research award mechanism, and the 4 prevention award mechanisms, and having conferred with CPRIT staff and SRA staff, our third-party grants administrator, I'm satisfied that the application and review process followed applicable laws and agency administrative rules, and I certify these award recommendations for the Oversight Committee's consideration.
Regarding conflicts of interest, Mr. Angeli has reported a conflict of interest with the awards recommended for the University of Texas at Austin. Are there any other conflict of interest declarations for the OC that have not yet been reported? We will vote to arrange the voting to accommodate Mr. Angeli's conflict of interest. Members, you have the list of applications and grant amounts recommended by the PIC for the Academic Research Grant Awards. The PIC recommendations will be approved if 2/3 of the OC members present and able to vote approve the recommendations. Dr. Wilson presented 6 academic research award slates. Rather than taking a separate vote on 6 different grant mechanisms constituting 51 grant recommendations, I'll ask for a vote to approve all of the slates. I note that application RR170064 that was recommended by the PIC was withdrawn by the applicant after the PIC meeting. Therefore, that award is not included as part of the awards that we'll vote on today. If a member wants to consider 1 or more award recommendations individually, please make a motion to do so now. Hearing none, we'll take up the University of Texas at Austin recommendation first. I'll entertain a motion to approve the PIC's recommendation for RR170054.
So moved. Second.
Have a motion and a second. All in favor, vote aye.
Aye.
Any opposed? Hearing none, and we have approval from 2/3 of the members present and able to vote. The motion carries. I note for the record that Mr. Angeli did not vote on this recommendation. I will now entertain a motion to approve the PIC's recommendations for the remaining 50 academic research awards.
So moved.
Second.
Have a motion and a second. All in favor, vote aye.
Aye.
Any opposed? Hearing none. Hearing approval from at least 2/3 of the members present and able to vote, the motion carries. I now would like to entertain a motion delegating contract negotiation authority to the CEO and CPRIT staff and to authorize the CEO to sign the contracts on behalf of CPRIT.
So moved.
Have a motion and a second. All in favor, vote aye.
Aye.
Any opposed? Hearing none, the motion is approved. And again, Dr. Wilson, I'd like to thank you and congratulate you and your team for putting together an outstanding slate of grants. Exciting time for Texas. Thank you. I now would like to recognize you to present the academic research programs plans for active and proposed requests for applications for FY18 and early 19. It's in tab 5.4 in your meeting book. And for the members of the Oversight Committee, you note that this is a new step as far as our process of generating RFAs and ultimately approving grants. Each of Dr. Wilson, Mr. Lang, and Dr. Garcia over the course of the year, and Dr. Garcia later today will present their RFAs to the OC for our approval, give us an opportunity to better understand the direction we're going with our RFAs, because those really do set the course for what we do as an agency and provide us an opportunity for input. So, Dr. Wilson, you're the guinea pig for our first run. Thank you for your work on this. Look forward to hearing your presentation.
Delighted. And if I may, because it is relevant to this program, if I could step back to a question, Mr. Guerin, that you asked about population disparities and cancers of importance in Texas, and recognize the important work that my colleague, Dr. Patty Moore, has done in putting together a summary of priorities, the impact of the priorities discussions on our portfolio over the past 3 years, and you will be looking at this later on. But just in response to your question, over the past 3 years, 17% of the number of awards that have been made have been in the areas of population disparities and cancer of importance. So
thank you.
Just an opportunity to advertise Dr. Moore's important efforts to help inform us, and I was a little slow in the uptake in answering your question.
All right, thanks for bringing that up.
So the purpose of that I took from the charge for this presentation was twofold. One is to provide the full Oversight Committee with the report from our subcommittee of oversight members who have, since I've been part of the organization, been intimately engaged in helping guide the RFAs that are presented to our applicants and then subsequently for applications. And then second, to review with you some of the important issues regarding the fact that we do have a limited number of dollars available for all of the good work that CPRIT's doing, and so we need to engage the decisions about RFAs with realistic budget considerations. So to this presentation, I want to begin by just reminding you of the academic research program priorities that we were charged with from you in 2017. Again, we've talked about these recently in this meeting, but just to reiterate, recruitment of outstanding cancer researchers to Texas, investments in core facilities, a broad range of innovative investigator-initiated research, and then targeted areas of investigator-initiated research to include prevention and early detection, computational biology, childhood cancers, and population disparities and cancers of importance in Texas. With that in mind, this table has 3 pieces of information. On the left-hand side are RFA mechanisms, both those that are currently announced and in blue, those that are proposed for this for the second cycle of this fiscal year. And then there is information about the anticipated budget that, based on prior years and our priorities and anticipated budget for each of these. As I think you appreciate, for the first cycle of fiscal year 18, we started this process 6 months ago, and in fact already have received 535 applications for individual investigator research awards. And shown here are an anticipated success rate based on funding of about 8%. We don't know what the success rate will be based on recommendations from our Scientific Review Council. It's ranged in the order between 12 and 15% in the past. I also want to point out that the recruitment RFAs are initiated each year and are opportunities for a monthly application from academic centers throughout the state.
I'm sorry, could I ask a question? Or I didn't know if
Absolutely.
you wanted to wait till the end.
No, please.
Please go ahead. I think it's
Trying to understand, did you just say the budget you have here anticipates an 8% success rate?
For the individual investigator research awards, we have 535, and what I'm presenting here is a budget that would support approximately 8%.
And that's below what our norm has been historically.
The Scientific Review, we have in the past been able to support all of the recommendations by the Scientific Review Council. As you know, today we had 11 that we were holding back, but to date we have been. The success rate based on the panels is on the order of between 12 and 15%. It differs between the individual panels and instruments, but about that.
Let me clarify something at this point. The budget numbers that you see on the right-hand side of this slide totals up to nearly $200 million.
It does.
The apportionment of the CPRIT funding across our different investment areas ultimately may or may not reflect this number. So when we vote later this morning on approving the direction of these RFAs, we're not approving this budget amount. This budget issues will be something that will continue to be debated, discussed among our professional staff, and we'll come back to the Oversight Committee. And as we all know in the Oversight Committee, this is a subject of ongoing conversation among the members of the Oversight Committee. So as we go through this, don't think that we are signing off on this right-hand column. This is just a way to put numbers and perhaps compare within his RFA total relative amounts for those different areas, but we are not approving or disapproving the amount of funding that will go into the research column over the coming year. So I just want to make that very clear. So.
If I may continue with the recommendations, because I think that Mr. Guerin raises an important point, both from the standpoint of your deliberations as well as your consideration of the proposed RFAs. So I appreciate that setup, because that's really pertinent to the four mechanisms that are described for the second cycle of fiscal year 18. Core Facility Support Awards, High Impact, High Risk. You heard about those earlier today, similar initiatives as we discussed earlier today. The Early Translation Award is a mechanism that has evolved from our experience with the Early Translational Research Award, intended to stimulate the development of moving discovery from an academic center into a commercialized entity. And with Mike Lang's leadership on this, and I think really inspired thoughts about how to accelerate the impact of this type of award on academic institutions, we've met in the past 6 months extensively with the technology transfer officers across the state, as well as investigators and our advisory groups, and come up with a strategy for an award which is intended to de-risk the discoveries in the hands of an academic individual that would set up a startup company success. And so that's what the intent is for the Early Translation Award. And then the Multi-Investigator Research Award are the program projects, the multi-project awards, which we did not have this year because of funding limitations, but because of advocacy both from the research community, our advisory groups, and the Research Oversight Subcommittee. We're bringing these back, and you'll see that we are anticipating a very small number, ability to support a very small number of these awards. They have the promise of building some of the cooperation across the state that you heard from Dr. Halton today in the example of core facilities. Like those core facility awards mechanism that he was mentioning, I think these are another mechanism for stimulating that kind of activity. They also are important in terms of our goal of getting the discovery into patients, either patients, clinical cancer patients, or in a prevention setting, because these awards typically have significant components of clinical trials. If you recall, last year, 4 of the MIRAs that were approved have major components of clinical trial research associated with them. Having said that, what we're presenting is an anticipated budget of as Mr. Guerin mentioned, about $200 million. This exceeds by $23 million the proportion of that's proposed in materials that had been provided earlier and reviewed by the subcommittee. So it's to Mr. Guerin's point that, quite frankly, it would not be possible to do all of this with less than the $199 million. And so part of our discussions with the subcommittee, as well as internally, is a prioritization. And what we're bringing to you is listed here as the program recommendations for priorities: recruitments, individual investigator research awards, core facility support awards. And if we can't support everything else, we will be very assured of having a very robust research program with those 3 mechanisms.
Mr. Roberts, would you speak to the budget issues? Because as we've discussed previously, and the Oversight Committee, it is a subject of ongoing discussion about how we allocate the resources among these grant areas. But you have pointed out that in every case we have been able to fully fund the recommendations. So could you speak to that quickly? I think that's important.
Certainly, thank you. If you'll recall, when we had our work session in May 2 years ago, a percent breakdown between academic and product development research was not agreed upon, but there was a stated intent that we fund more product development than we had previously. Historically, the split between the two runs about 80%, 20% being product development. Over the course of a couple of years, I set targets internally that would get, it went to 25% product development, then to 30. We have been unable to get to the 30% target for product development. When that occurs, by default, the remaining funds allocated for product development have gone to academic research. And historically, either by that transfer or deferring action on certain grant awards to a September meeting. I don't think we're going to have a September meeting this year. We have been able to fund all of the projects that have been recommended to us by the peer reviewers and to you by the Program Integration Committee. Another mechanism that's been used is evaluating the actual budgets of the proposals with some trimming, which occurred earlier today, without damaging, we think, the viability of the project. So looking down the pike here with a year we haven't even entered this fiscal year, I am not overly concerned about our ability to fund any of the proposals that will come to us. I am a little bit concerned in the case of product development that we have not in the last 2 years received a sufficient number of applications that have made it through the peer review process. But that said, I think it's a testament to the peer review in product development that they're not just going to propose something in order to fulfill a funding quota. And it's up to us as an agency to try and beat the brush and get a greater number of product development awards. But that has allowed, anyway, for the last 2 years, academic research to exceed the amount that was proposed.
Thank you, and you have warned us, and I know it's something we're all mindful of as OC members. We don't want any sort of quotas to get us to do anything other than spend money where we can have the highest impact. And we will continue to have the policy debate among OC members about emphasis, but at the end of the day, we're going to fund the best opportunities where we can make the greatest impact. Thank you. Mr. Montgomery?
Thank you. Dr. Wilson, on the Academic Research Program RFA recommendation for FY 18-19, the 3-page handout, on the very first page, you've got 7 different areas, the last of which is population disparities and cancers of importance to Texas. But on the next page, that category is missing. Can you tell us what's going on there? You run out of room on the page, or?
No, that particular element of the priorities is incorporated in all of the RFAs and represented, and as mentioned earlier, successfully in terms of the number of awards being, whatever I mentioned earlier, being 15% over the past 3 years.
Okay, thank you.
All right. Any other, Dr. Rice?
Thank you. I wanted to just highlight maybe briefly a bit of the detailed discussion we had during our subcommittee, the research subcommittee, academic research subcommittee, just for the benefit of the Oversight Committee. The question at hand, and Dr. Wilson may want to restate, or I'll characterize what I think I heard, and then I'll let you correct if I missed it. But there is always concern that you don't want to have an RFA put out there, and you don't want to have researchers across the state spend dozens, hundreds of hours, whatever, building 100-page documents for applications, and then all of a sudden have either a very low or just close it all off and say, well, we don't have any funds for this kind of an RFA. So clearly, that's, I think, a concern that has been expressed. Just to say, maybe if we didn't have a full budget, you wouldn't go ahead, you would cut off some of these last 4 RFA types, and that might be how we proceed. And the reality is, and this is the conversation we'd had, that we've ended up with the bandwidth to fully fund and go ahead and never close down, or maybe actually we held Myra's 1 year, but the point is that we want to largely put these all in the field and have these RFAs in play this coming year, 2018, and the way we can anticipate accommodating, even though we might not say this is a firm budget, but the way you would get there, of course, is the things we've already done, which is to say, sometimes we roll things over, and sometimes we delay things just to watch and see how these applications and how the budgets come through. In the year 2017 fiscal year, of course, it turned out that we didn't fulfill the product development. The product development volume of dollars that came through and got approved all the way through the PIC and all has been actually 14%, I think, so not 30%. So I guess what I'm trying to say is that through various mechanisms and also the reality of what has come through, we've never bumped into the ceiling and never been able to, we haven't had to unfund or not fund one of these. And so I apologize if the eloquence is low here, but the point is that, my sense is that, and this may not be where we want to fully develop the conversation, but my sense is that we should, plow forward with all of these, knowing that historically we've never hit the ceiling. We've always had wages to manage around if we were close. And this year, product development, didn't hit 30 and didn't try to hit 35. It actually hit 14%. So it was lower, not higher. So there's no reason to anticipate that we'd, have a massively different outcome in the future years necessarily. And if we did, then that'd be fine. We'd have to deal with it. But I'm just putting forth the point of view that we certainly don't want to think about 2018 and say, let's not do multi-investigator awards because, geez, we might not have a budget. That would require us to assume that none of our mechanisms have worked in the past and that the recent history of the last 2 years actually would not be what we'd expect to be repeating, if that makes sense.
Thank you for that clarification, but I guess the point I was trying to make earlier, we're not locking ourselves into a $199 million commitment in this area, and the staff will work among themselves to adjust as opportunities present themselves. It won't be exactly $10 million in early transition. We're talking about not rough, but estimates at this point that Mr. Roberts will work within the organization, and he has successfully in the past to make sure we've had the funding available to fund all of the grants that have come to us as recommended. So I just want to make sure that nobody thinks we're signing off on $199,600,000 worth of grants. We're signing off on the left 4/5 of the page, if we, in fact,
Thank you.
do vote yes.
Thank you.
Yeah.
A few questions. First, Jim, are you done? Have you completed your presentation?
I have not,
Okay.
but welcome the interruption because it's been very helpful.
Maybe it'd be better if you went through finished your presentation.
Yes.
first.
So, so the next slide is really to bring to a visual of this Gantt chart what this mechanism looks like. And I won't go through the details, but as I mentioned to you, the red is already in progress. The very top line is the recruitment process that takes place every month and then is reviewed by the PIC and Oversight Committee on a quarterly basis. We are proposing to carry this on into fiscal year 19 next year. The 18.1 is the investigator-initiated grants that I have mentioned that have already been submitted. So we're at the point of having received 535, and they will be reviewed in October and then presented to the PIC and Oversight Committee in February. That same process we're proposing for the core facility and high impact, high risk, and perhaps the other mechanisms to take place over fiscal year 18. And you'll see that those need to be published for the community to respond essentially tomorrow. And then the grants will be prepared and submitted in January, reviewed in April, in June, and then presented to the Oversight Committee next August. So you see that we're working in a one-year sort of life cycle of these mechanisms, and that was the intent of this particular Gantt chart. Dr. Rosenfeld, I'm completed my presentation.
Thank you. A couple quick questions. When someone submits an academic grant, is there a pre-application process where they submit a short summary, and then those are called down, and the good ones where you send a full application?
There's not. There is, for the individual investigator research awards and the recruitment, these are reviewed by our review panels and through a process of two-tier review. A quick assessment of culling out from that 535, approximately half that would then get a more extensive review. But this is after the submission.
I was just wondering.
In the context of the high impact, high risk, MIRAs and core facilities, those all get a full review, but they've been, if you will, vetted by the individual institutions through a mechanism that limits the number that can be submitted by a particular institution.
I was just thinking of Bill's comments about investigators filling out 100-page applications. Whereas, for a lot of grant processes, you first fill out a pre-application, and then there's an invitation for full applications. I wonder if that's a good idea or a bad idea. I mean, it's a matter of saving, if someone's not going to be funded, it saves them all the work of taking weeks or months of their life to.
Personally, I think it's not a particularly tractable idea from the standpoint that it's very hard to figure out how to actually vet those unless you have a very proscribed criteria for them. And these research awards are investigator-initiated, and so, again, really, that type of process isn't as effective. However, having said that, we recognize the issue that you're raising, and for that reason, we do charge the institutions with helping to vet these, and so that hopefully there is a screening process for several of these larger grant mechanisms, such as the core facilities.
Okay. Thank you. I'd like to talk about the Early Transition Awards and the MIRA Awards. And I think probably everyone in the room knows my opinions on this, but just to repeat myself over and over again. On the Early Transition Awards, I'm very strongly opposed to these type of awards. And I'll give you a few reasons, but I think these should not go forwards. Some of the reasons, and I just wrote these down as you were giving your very nice presentation, is the overriding reason. I don't think this is something that CPRIT should be in the business of funding. I think these type of funding should come from the institutions itself, and what we are doing is doing the institution's funding job for them. These institutions have lots of money that they could provide, $1 and $2 million grants as forms of seed grants, and I think that overall, I think that is the job of the tech transfer offices and the universities to provide these funds, and not CPRIT to provide these funds. Theoretically, I don't have a problem with the concept, if you will. It's just I don't think that's a CPRIT responsibility or a good expenditure of CPRIT funds. To take that a couple other steps further, the Early Transition Awards, as you described and we've talked about before, implies that academic researchers know how to do preclinical drug development. And I can tell you from being on both sides of it, from being as a pharmaceutical company, as being as a clinical academic researchers definitely do not, although they think they do not have the expertise to do preclinical drug development for INDs. It just doesn't exist. And that money, I'm convinced, will be flushed and go away. Another reason is, and I know you know all this, is that a couple years ago there was several publications on how poorly pharmaceutical companies published results of clinical trials, and it was a lot of papers were published on that. But then someone asked the reverse question: how successful are academic institutions in reporting the results of clinical trials? And actually, academic centers were much worse in reporting results of clinical trials, and in fact, more than half of them were never completed. For lots of reasons. As you know, academic researchers leave or go to other institutions, lots of reasons. So I really think that these Early Transition Awards are not the business. I think we have a low chance of success. I think this is the responsibility of the institutions if they think it's worthwhile to invest in them. So I stand opposed to the Early Transition Awards, and I'm glad to engage in more conversations with you and Mike and others about this. On the switching over to the MIRA Awards, just a couple small comments. One is you have an exception for the Early Transition Awards that excludes public or private companies for the Early Transition Awards. But for the multi-investigator research awards, you do not have that exception. And I was wondering, so if a pharmaceutical company wants to come in and apply for AMERA, could they do that as opposed to going through the product development route?
No, this is an oversight that that's not represented in the materials in front of you, but is in the
Okay.
proposed RFA,
All right,
and
thank
has
you.
been in the past. Yeah.
All right, appreciate the clarification. And the other, on the Multi-Investigator Research Award, I may not be reading this correctly, but to me, this, for clinical research type studies, certainly sounds like what you're trying to do is set up a cooperative group. And as you know, the federal government funds cooperative groups, and I'm wondering, am I reading that right in the realm of clinical research? Are we setting up a Texas cooperative group to enhance or compete with SWOG or ECOG or Children's Oncology Group?
No, we have not communicated in this brief synopsis the true intent of those awards. It's not to set up a clinical trials organization. It is to facilitate interactions between investigators with complementary expertise, one component of which might be the ability to translate into early phase clinical trials a novel idea. So, for example, last year, this group approved a very innovative MyRA-2 MD Anderson and Baylor College of Medicine targeting acute leukemia. And as part of that, MyRA were clinical trials which were bringing into patients early precision therapy targeted trials and immunologic-based trials that were based on the discovery in those complementary programs. So it's the sorts of academic institutions, early phase 1 and 2 trials that are being supported here.
Okay,
Yeah.
appreciate the clarification. That really helps. So as it stands, I would have to stand opposed to the early translational awards. I just don't see them as the stuff of CPRIT. Thank you.
Mr. Guerin, may I make a comment?
You certainly may, and let me just,
please.
we're building this airplane as we fly it. I said you're our guinea pig. Should have said our zebrafish, just to be very current.
I'm transparent, is that what you meant?
And what I would like to propose to the committee when we vote on these, that any one of these issues that I anticipate that we're going to have approval of most, if not all of these. But if anyone would like to have one of these pulled out for a separate vote, so not to be put in a position of having to vote for all or none, I'd ask the OC members please make that request. And in order to accommodate Dr. Rosenfeld, we will, I recommend that we vote separately on the early translation award and then consider the rest as a slate unless someone asks that a category be voted on separately. So as we listen to this discussion, OC members, just please be mindful of that as an option. So you don't have to vote to take the whole thing down if you find yourself troubled by one aspect of it. So, Dr. Wilson, please go ahead.
Only a brief comment to say that this has been a very instructive process that the CPRIT staff, and particularly the research program, has been engaged in over the last several months in thinking through the RFA schedules and over the next several years. Our subcommittee discussion I found similarly very helpful and informing. I would like to suggest that this discussion today is similarly very helpful to the research program as well as, I hope, to the Oversight Committee members, and want to request that we continue to have the opportunity to have these exchanges because they turn out to be very helpful to us in sort of taking advantage of your oversight of over the past, collectively your oversight over the past several years, as well as your view into the future. And we need this interaction with you to help guide the program successfully.
Well, thank you, Dr. Wilson. I think I speak for the whole Oversight Committee when I say that we've deeply appreciated the opportunity to be full partners with all of the professionals that we have on the CPRIT team, recognizing that our role is different than your role, but our goals are exactly the same. And I feel that we've had that sort of spirit of partnership from the very beginning, and appreciate that very much. In that same spirit, following up on Mr. Montgomery's question, you know, one of the purposes of having this involvement with the RFAs is to get a sense of where we stand going into the next year or 18 months on our program priorities. And as you answered Mr. Montgomery, the cancers of importance to Texas and disparities are embedded within these categories. Could you suggest to me how we could, going forward, provide information to get a sense of, you know, how much is embedded in those, the categories that you have listed there, so we have a sense, does this mean, you know, a little, a medium amount, a lot? And perhaps it's a question of presentation at this point, but what sort of guidance do you have embedded in those categories that is going to encourage RFAs in those two areas, and how could we present it so we have a sense of that?
Well, as I've mentioned, there's been really two components of how to guide the community in terms of responding to the priorities. The first is to actually publish RFAs which target a request for applications, and specifically those are identified as you've noted. And I would argue been highly successful. When you look at some of the information you'll review later, childhood cancers is...
Yeah, my question's a little different, though, is based on the presentation you're making to us today and you will make in the future on RFAs, what could you include as part of this presentation so we get a sense of what the emphasis is going to be in those two areas? Obviously, not every one of these is going to have that emphasis, but help me understand, or maybe help us develop a presentation that could somehow quantify, and not rigidly quantify, but quantify what your goals would be in those two areas, and how you could represent that on this slide in some way, so we have a sense that when we're voting on this, we know that, you know, 30% of these are going to be going to encourage RFAs in disparities or cancers of of importance to Texas. What could you tell us that gives us a sense that that number is 0 or 30%, 50%, 90%? How could you communicate that?
Well, I'm not sure I can say more than I already have in terms of the impact to date.
I'm not talking about impact to date. I'm talking about how you inform us today looking prospectively. And maybe I'm missing something, but I have no way of knowing from looking at this how much emphasis there's going to be on disparities or cancers of importance to Texas.
Right.
And so that's what I'm trying to get at. I'm not saying that it isn't going to happen, but I don't see how I can glean from this how much of that to expect and be able to say, well, we hit our mark, we missed our mark, right, and allow us to continue to be thought partners in figuring out how we are successful in those areas.
Thank you. I think I understand. I think it is possible to proscribe a targeted RFA in that area if one reviewed the data and saw a deficiency in the portfolio. That was what you did before, and that's what I think would be the answer to your question, that to date we haven't seen that deficiency in the portfolio. So to be very specific, I think to answer your question, if you felt that the portfolio was deficient in an area, a targeted RFA in that area would be, it has proven to be a very successful way of increasing the representation in the research portfolio.
Maybe I'm just not fully understanding what you're saying. The past practices have yielded results that we're satisfied with. I think that's one. I think we're all in agreement on that.
Yes.
And I'm just trying to understand. You're saying based on those past practices, we ought to assume that this is going to yield equally satisfactory results.
Well, I think also taking a look at the track record and areas that you are interested in as priorities, and if those are not satisfactorily, or they're not represented in the portfolio, then a new intervention would be appropriate, such as a targeted RFA.
Well, why would, for example, we put childhood and adolescent cancers on here specifically, but we don't have cancers of special significance to Texas? Tell me why one would qualify for a category and one wouldn't.
Yeah. Well, I think that decision predated me, but I think it was the right one because those areas extend across all of these mechanisms and are contained within the RFA as an area of priority interest for CPRIT. It turns out that the portfolio, as I've mentioned, is robustly represented in those areas, my opinion. But if it's not satisfactory, then one approach would be to add a targeted RFA if that was.
Why do you have childhood cancers as a category, but you don't have cancers of particular interest to Texas as a category?
Because that was the area that was deemed at the time to be underrepresented. Similarly with computational biology and prevention research, those were all, under Dr. Kripke's review, underrepresented in the portfolio.
If I could expand, Mr. Guerin, I think I understand the point of your question. And I'm not trying to kick the can down the road, but the program priorities discussion in November can identify that, but that we ought to do that. But specifically, the way you can do it would be similar to what Dr. Garcia is proposing in the prevention program, is that for a perhaps one cycle or two cycles, that we restrict the RFA to disparities or to cancers of particular interest to Texas and to computational biology and the others, and you limit the amount that you receive just for the general interest research applications.
I think that's a good approach, but I guess one thing you said there at the end, Dr. Wilson, that you inherited from Dr. Kripke, those two priorities, computational biology and childhood and adolescent cancers. Since that time, the board developed priorities, and I don't think that the board said those two were of more importance than cancers of special significance to Texas. Regardless of what Dr. Kripke had left to you, since that time, we came up with our priorities that this Oversight Committee voted on, and I believe that we didn't weight them. And so the way you described it, that these were just holdovers from Dr. Kripke, there's been an intervening decision by the Oversight Committee which added those other two categories. So if it's only because that's the way it was done under Dr. Kripke that you present them this way, I don't think that holds for today.
Let me just, if I may, interrupt and say that I didn't express accurately what I think has happened here. It wasn't that we inherited a set of RFAs from Dr. Kripke.
Not RFAs, priorities.
Well, priorities and RFAs. My point was that Dr. Kripke, you, identified childhood cancer and computational biology as a priority, and the intervention was actually two things, and I only was able to mention one: the RFAs that were targeted, and then second, constituting review panels that were adequate to review them. And that was what happened in response to priorities, the priority assignments in the past. I was using that as an example to address your question. So if you review the portfolio and deem it deficient in a particular area, the approach that has had a track record of success is the development of a targeted RFA.
May I
Sure.
jump in?
Please.
I don't mean to interrupt. But Dr. Wilson, correct me if I'm wrong, but it seems like to me that if you release an RFA in a generalized area, individual investigator prevention or clinical translation core facilities, you're going to capture a certain amount of the cancers that are of particular interest to to us or to Texans or disparities. But childhood cancer and computational biology are different in kind than other areas. And so you have to go direct the RFAs to them specifically, whereas if you direct a general RFA on a subject, you will capture, of necessity, more liver cancers, more cervical cancers that are disproportionately represented, by example, in Texas. Is that accurate or not accurate?
Well, that's what the data showed. And that was the point that I was trying to make, is that if this can be a data-driven discussion and inform whether a specific intervention is necessary. I think that was the point, at least attempting to answer your question.
Can I make a comment? Thank you. I was just thinking out loud here. I will think out loud, I guess. You know, we also, it seems like might be having a discussion that is a little bit circular, so let me see if I can do it. We might say we want to have priorities of cancers with focused attention and funding of cancers of importance in Texas and in cancers of disparities, that et cetera, in this. And we might say, let's say we want to spend $40 million a year to do that, and we'd spend 1/6 or 1/7 of the dollars we spend for research, let's just say 1/6, and we might all say that's perfect and that sounds about right for focusing on disparities and focusing on cancers of special significance, given that all the cancers that we're studying overlap largely there anyway. I mean, breast cancer affects people with disparities. Lung cancer, pancreatic cancer, all of those things affect people with disparities. So what I'm getting to is that when I look at the numbers in this table and multiply them by 2, we're probably at 40 million a year or just short of that anyway. So we're probably, if we took away the conversation and talked about how much should we focus on just on disparities and just on cancers of significance in Texas, and we said, geez, $40 million ought to be about what we spend and think about just for those, we already do that. So in some ways, I'm not sure we need to amplify this. It's something that's already actually happening, is kind of how I look at it. So I want to lay that out there to the group. And I have another comment on early translational grants. But I'm just wondering if we don't already do what we're trying to do. And, you know, you don't want this to be 80% because that would make no sense. There's, you know, the trouble of cancer in Texas is population-wide. And actually, I think we're largely doing what we want to do, but based on the numbers that we look at currently.
Appreciate that.
One other short comment. It seems like what Mr. Guerin is talking about, how do we prospectively know we're going to hit the targets for cancers of importance to Texans, for instance? And we know historically we've done it. Your suggestion is, Dr. Wilson, if we continue, we're likely to continue that hit rate, but if we don't, then we correct it in the future. I just want to add a small footnote, and that is that the institution was started with the goal of funding the best science. If you direct it toward a particular problem, you run the risk of moving away from that goal. I'm not suggesting that we have or we will, but there is some risk there, but that's it.
Mm-hmm. And that's something we've borne in mind from the very beginning. Right. And with that in mind, still came up with these priorities, with asking that they never be followed to a degree that they would compromise getting the best. So I see what you're saying, is that we have been able to address those priorities without identifying them specifically. And whether that's a reason not to put them on this list in some way and hold them for course correction later. Again, we're building this as we're flying it. And I thank you for this robust discussion on this point. And then just ask you, and I'm comfortable with where we stand now, but the purpose of this exercise is to engage the OC early and have this discussion to ensure to the best we can that we advance our grant making along the lines of our priorities. And so you've helped me understand why it's not on this list. Perhaps going forward there might be a different way to present it, but I'm comfortable that our past record is going to ensure that we do okay in those areas. But I would like you to think about how you could inform us at this stage of the RFA approval process that we can vote in favor of this, comfortable that those priorities are going to receive proper attention. Okay.
Could I speak on early translational? Are we,
Please do.
If we're okay. I want to add a very amicable and symmetric response to the early translational comments that my colleague, Dr. Rosenfeld, had made. And that is to say that we've thought a lot about this. We've worked a lot with Michael Lang as well as Dr. Wilson, and it may be just a matter of opinion, and it may just be sort of part of the experiment of this, of this process and all. But my sense is that the early translational efforts to fund small companies to help hatch out ideas into new companies, is a very important and viable use of the funding that has, that CPRIT does. It's again one of these quote valley of death things. There's a couple of valley of deaths from an idea for a new kind of molecule or whatnot to get to a patient, and one of those valley of deaths is actually formation, new company formation. And as we heard from, I think, Mr. McQuitty, I think, last time, there's another valley down the road where they need to raise $20 million, and we've been attending to that valley somewhat as the as the approach that CPRIT's had in the commercial product development, commercialization kind of realm heretofore. So I have liked the idea of an early translational RFA that hatches companies out to get them across the first valley of death, because to me, you get 10 shots on goal, so to speak, with 10 times 2 millions versus 20 million funding, whatever, as we've done. So that's how I look at it. Now, all the things Dr. Rosenfeld discusses about the pitfalls of people needing the right expertise, and a generic professor doing research is not the same as a generic CEO of a biopharmaceutical. So I think you have to build those things in, and those checks and balances as parts of milestones and all the things you would do to make sure that as things get funded, you work them. And probably not, this is maybe the whole conversation needs to come to a special, meeting where we talk for a whole day about priorities. But I have a strong sense that 20 shots on goal is something we should be thinking about as we balance that with the history of our organization, which has been mostly in the commercialization product development side, been shots at the 10x times, instead of having $2 million, we've had 120 or sequences such as that. So
Okay.
thank you for
Appreciate that. Let me just make a clarification that got some advice from my counselor, sits to the right, but counsel to the left, that when I was referring to flying the plane as we build it, I'm talking specifically about this, the OC being involved in the RFA process at this stage of the game. Mr. Montgomery was concerned that some might have heard that as referring to the CPRIT mission overall. I was merely talking about this one exercise we're going through today, but sometimes lawyers listen too carefully, but I appreciate that he did. Dr. Rosenfeld.
Good lines, Pete. To ask Bill a question or two, I absolutely agree with your approach of more smaller grants to seed companies. But if I look at the early translational award, I just don't think that's the mechanism to do it. Among other things, it says that companies cannot receive the funds. So we're not sponsoring multiple shots on goals for startup companies. We're sponsoring universities. And to me, that's a huge difference. But I agree with what you said philosophically, but I don't think this is the mechanism to do it.
Is that a question?
Of course.
Thank you. Yeah, I don't actually agree with that part, and that's not my understanding of it. So I'll reserve the right to comment later, and we can discuss this mechanism more as we go through a priority conversation, because my thought is that is exactly the opposite. It would be for a formation.
There actually is two parts to the proposal. You're only hearing the academic research part of it. The product development research of it is for nascent companies. That's correct?
Yes, it's a tandem, and yes, it's a tandem.
This is 50% of the program. I'd also like to point out, Dr. Rosenfeld, that on the Gantt chart that this RFA would not be released until April, and I think that you raise some interesting questions that it's incumbent upon us as staff to be able to respond to, and that in the ensuing months, whether, I mean, if the Oversight Committee approves the concept of this RFA, that we work with you, because I have a sense that your insight could improve whatever RFA we put out. And so we would like, I would like to send a delegate. I'll volunteer Dr. Wilson and Mr. Lang and myself to meet with you and address some of your ideas.
There we go. I wonder if it's a worthwhile experience to getting those, the people on the Oversight Committee who have an interest in this, and the staff of CPRIT who are interested in this, and work more on this type of an award and bring it up at the next meeting instead of voting on it now.
We're not voting on an award now.
Well, are we voting on RFA?
The committees will have an opportunity, subcommittees will have an opportunity to engage on the details. But yes, we would be voting on this as his RFA plan. Yeah.
I'm wondering, as far as the early translational award, if we could set that aside for a later meeting until we've had more discussion.
Let me propose a procedure that would allow you to amend the proposed motion when we get to the early translational, because I plan to set it out for a separate vote, and then you can offer that up as an amendment to the motion, if I could. All right, any other questions? I appreciate very much the opportunity to have this discussion. And as Dr. Rosenfeld raised his objection to the early translational awards, where we will have two different votes. First, we'll vote on Dr. Wilson's proposal plan for FY 2018 and early 2019 RFAs, with the exception of the early translational awards. Do I have a motion?
Mr. Chairman, before you do that, did I understand Mr. Rosenfeld, though, to say he actually supported what it sounds like this was intended to say? In other words, an early translational research award actually is an award to a company.
Could I ask you to discuss that when we get to the vote on his? We're going to vote on the early translational separately. So this is on everything except that. Is that all right? So we'll have an opportunity to address that. Do I have a motion to approve all except the early translational awards?
Second.
And a second. All in favor?
Aye.
Aye. Any opposed? The motion is approved. Now we will move to vote on Dr. Wilson's proposed plan for the 2018 RFA for early translational awards. Do I have a motion to approve? Dr. Rosenfeld, would you like to propose an alternative motion to that?
Please, thank you. I'd like to propose that the Early Translation Award be further discussed between the Oversight Committee and CPRIT and brought to a vote at a subsequent Oversight Committee meeting.
Could I ask the staff to comment on that, considering the calendar that's in front of us? And
Could I just make a couple of points to that? First, the Early Translation Award is to academic centers to de-risk a discovery, lead compound, by that to conduct the toxicology, et cetera. And it would be packaged with a product development award, which is the seed award. So that's the tandem that I was talking about. I personally think that, and I don't want to speak for Mike, he should comment at this point. I think both of us would welcome this discussion. I think this is a very important discussion and one that we would very much appreciate the expertise both on this group as well as others that you may wish to engage with us. So I think that would be welcome from my point of view. Mike?
Yeah, we've had.
Whoa.
Well, he said staff.
yeah, let's try to restrict this
Come on up here.
discussion to the
And I have a question
his,
before we vote.
amendment to our motion.
All right, or,
All righty, Dr. Rice?
well, I was just going to ask, what's confusing me a little bit is that I think of the Early Translational Award as a specific award that we've talked about in the context of maybe having two parts. There's a part that's on the academic side. I guess I hadn't had clarity on that. And then there's a part that would go to the new company that got formed. That's actually one package. And maybe there's you get off the bus if you flunk the test because of some toxicology.
Is that
work.
one
So-
package? Could you talk through that?
Mike, help me out.
Well, this has been a subject of ongoing discussion for some time. It was discussed in the Product Development Subcommittee meeting, as Dr. Rosenfeld recalls. At my tenure at CPRIT, I've been kind of struck by the fact that we fund academic research and we fund companies when they are off the ground, up and running, operational stage companies. And we'll be presenting a company today that's in that phase. But we don't really focus on the intermediate steps. So I think CPRIT's mission is advantaged, is optimized by us providing a continuity of funding through all the stages of the development process. Today, we fund, basic and applied research. The substantial slate of proposals were presented today, which are seemingly well received. We then do not specifically fund, as Dr. Wilson points out, the de-risking of that. Here's an interesting finding. This particular compound, for example, seems to have novel value and could be the basis of a new therapy. But there's questions that need to be answered before it's an investable compound. And the awards that we're discussing would address those questions and would de-risk that compound. So that's one set of mechanisms that have been discussed and would be housed in the research program, because those funds would be provided to the academic institutions. When that work is successfully completed, not all of it will be, but when it is, then you have an opportunity to start a company, and we are hypothesizing or proposing another award mechanism that would facilitate company formation and company operationalization.
So are you envisioning that as a separate RFA, a separate application process?
That is correct. That's what's envisioned. And again, we can have these discussions, but...
The reason I have trouble with that is, of course, one is directly linked with the other. We'd never have the second without the first. So we're right now having a conversation about wanting to delay the, sort of the consideration of this first thing, but it only makes sense when they're actually coupled and considered as one.
I think in many cases there would be a continuity of CPRIT funding, but recognizing, for example, today we are not presenting the seed award proposal, but we'll probably be presenting that in the next meeting. Well, there may be companies that are projects that are ready for a seed award, but because they're just because that mechanism is just now available, they would not have previously received, a translational award. So we don't anticipate coupling them necessarily, that there's a one-to-one correspondence. You can only get money at step C if you got it at step B.
Does that delay things for a company that would have a process? They'd have to wait for an RFA to be accepted
not
and
at all.
funded,
A
and
company-
then they'd have to sort of have
We'd have
another
these
one?
various mechanisms, and companies are at projects, to be more specific, because some of them are pre-companies, but projects are at various stages of development, and they could enter this continuity of funding at any point. Certainly the ones that enter it now early then could progress through the various stages.
Is the timing, though, from start to finish of that like a 2 or 3 year process of applying, waiting for a year to get the initial funding, having some results, and then applying for the second part?
there's
So it'd be like a 3-year process?
Yeah, there'd be a several-year process
So
there.
I'm just
typically.
wondering if decoupling it unnecessarily delays progress.
No, I think it's the opposite. That coupling it would preclude any seed awards from being affected until 2 or 3 years from now, until after the translation awards were awarded, that were received, and that work was conducted and shown to be successful. So we essentially would be delaying the start of the seed award program by several years if we coupled them.
Could I interrupt for a moment, please? This conversation, this discussion has moved me from planning to vote no on Dr. Rosenfeld's amendment to our motion to move me to a yes vote. It does sound to me as if there are some things that we need to talk through before we understand it well. And so I would like to ask the committee to consider moving ahead with the consideration of Dr. Rosenfeld's amended motion, which would delay consideration of the OC vote on the translational research RFAs. And is looking at the calendar, Dr. Wilson, it does appear that this could be brought back to the OC in the November meeting without in any way compromising your calendar. You could continue to plan, but you wouldn't have to release that RFA until, I think, August of next year. Is that the?
So I think that we're very comfortable with continuing this discussion because I think it's an important one, and I don't believe any of us have hit the spot that we want to be. This will delay, but I think appropriately until we have the right mechanism.
Okay.
The
Could I
only
ask
thing
for a
I'd
motion?
say is I'm not sure it will delay it. It may, but I think that the schedule, the calendar can accommodate this discussion.
Dr. Rosenfeld, would you like to make a motion?
Make a motion to delay a vote on the early translation awards, and between now and the next Oversight Committee meeting, there'll be more discussion between the Oversight Committee meeting and the CPRIT staff.
All right, do I have a second? I have a motion and a second. All in favor?
Aye.
Aye. Any opposed? The motion carries. Thank you. I'd like to call an audible at this point and recognize that it's been a long time since some people have eaten. But we have our auditors here, and rather than ask them wait through lunch, I'd ask Ms. Martin, and I believe she's accompanied by Mr. Gray. And I'd like to recognize them to discuss 2 internal audit reports over the pre-award grant management and purchasing and P cards, as well as the follow-up procedures for 3 previous audits with their findings. And after she completes her report, their report, the Oversight Committee will move into closed session so that Ms. Martin may present the follow-up procedures over the prior year information security audit. Ms. Martin, Mr. Gray, thank you for joining us today. Appreciate it.
Good afternoon. Will you please turn to tab 13 in your meeting materials? There is an extensive summary of our progress for internal audit since the last time we met with the Oversight Committee. However, I will be able to brief us through the results. As well, we have already met with the audit subcommittee and talked in much more detail through each of the internal audit reports that are included in your package.
Right. Sorry, Ms. Martin, I don't mean to interrupt you, Mr. Chairman. As one of the longtime faithful members of the audit committee, I would love to hear the entire presentation again, but given the time and all we have to do, if you could shorten it as much as possible, that would be great.
Yes, sir. To that request, I will hit the highlights as well as the nature of the findings, and then point us to where we're going for fiscal 18. In 2017, we had approved, there were 4 internal audits that were approved to be conducted. We have completed all of our procedures on all of the fiscal 2017 internal audit plan that was approved. The first 2 reports on page 13-2 of my summary have already been briefed with the Oversight Committee. You have a report in your package over the pre-award grant management. That report, if I point you to page 13-13, you will see the executive summary of that report. This internal audit covered a very large and very important part of your grant-making activities. The report identified only 3 internal audit findings from the entire scope and received a satisfactory report rating. Those 3 findings are, 1 was high, related from an error, just a simple mechanics error coming from a spreadsheet when a grant award was changed, a value was changed. There was 2 moderate findings, one related to post-review statements. There was 1 post-review statement that had a signature missing related to 2 out of 40 applications reviewed, and then some segregation of duties issues related to access to a very small portion of the confidential materials in the SharePoint portal. Those 3 findings, you have a management's response, and we are comfortable that management is in the process of remediating those findings. Moving to the next internal audit, we did conduct an internal audit over procurement and P cards. That was an extensive, pretty wide scope. If I'll turn to page 13-31 in your package, you'll see that through our scope, we identified 9 findings. No high-risk findings. There were 7 that were moderate and 2 that were low. Of those findings, we were comfortable that the procurement process still covered the function and responsibilities and provided a satisfactory report rating as a result of our internal audit activities. Those findings are all related to what I would call the basic blocking and tackling of procurement documentation, and did not relate to any specific compliance issues, and therefore that is why you have a satisfactory report rating. All of those findings, you can see how they fall into the cycle on page 13-36, which should give you comfort that the procurement and P card, which is primarily a travel card program at CPRIT, has established processes, procedures, and controls. Yes, we did find a few things that could be done better, but there's an extensive amount of coverage that you can see on this chart at 13-36 from a procedures and controls standpoint. And we're comfortable that management is taking the findings seriously, and will look at the remediation activity in fiscal 18. Shall I move to the next reports?
Unless, any questions on this first report?
And the audit subcommittee, feel free to add, we talked about each of these much more specifically. Mm-hmm. We did perform 4 follow-up procedures. We're going to talk today about 3 of the follow-up procedures that we performed, and I'm happy to say that the procedures resulted in closing out all of the findings that were previously identified in fiscal 2017. 2016 internal audits. The first, I would have you turn to page 13-71. You'll see we had originally 2 findings that came from the revenue internal audit. Those 2 findings have both been fully remediated, and there's no further action. We also issued a strong report at the conclusion of closing those 2 findings from our follow-up procedures. So the revenue internal audit is completely closed. The next audit was over cash management, at which we identified 1 moderate risk finding in 2016. I'll have you turn to page 13-81. And at page 13-81, you will see that we were able to close that prior finding on page 13-82. The procedures are in place, and we verified such and provided a strong report rating on cash management. The last internal audit follow-up we performed was over the commodity and service contracts internal audit. That audit, we had 5 findings. Majority of those findings were on commodity purchases, but we performed follow-up procedures over all 5 of those findings, and that is represented on page 13-92. We were able to verify that 4 of those findings, new procedures had been put in place and had closed. 1 of those findings, specifically the one for commodity like supplies, purchases, not having a second review, was closed by management under comfort of their existing process. And we, as internal audit, agreed with that. So all 5 of those findings were closed. Results of the follow-up procedures and the fiscal 2017 internal audit plan, I would have you turn to page 13-5, which is our tracking schedule for internal audit status and findings. In the middle of that page, the 3rd block says fiscal 2017, and you'll see that you now have received 4 internal audits for fiscal 2017. 2 reports are in your package today. The findings that resulted from those audits will be included in the fiscal 2018 internal audit plan for us to verify management's corrective action. Further, our procedures to verify corrective action over 3 of the 2017 internal audits resulted in all of the findings closed and fully remediated. The 2016 internal audit over information security will brief the committee on separately, and that's the only remaining items open. Next, I'd like to have you turn to page 13-100. 13-100 is the start of the CPRIT fiscal 2017 annual internal audit report. This report is regulatorily required that internal audit prepare this report on an annual basis, communicating all of the audit activity that occurs on behalf of CPRIT. This report is in draft. It has been reviewed with the audit subcommittee and will require approval from you today. The first sections communicate our internal audit plan at 13, page 102 and 103, for which you have been briefed the results of. Page 13103 at the bottom begins the communication of audit work performed by Grant Thornton and BFMS as they perform oversight for the third-party observer reports as part of your annual requirement. And that extends for several pages, communicating their responsibility for those third-party oversight reports. Then on page 13105, communicate the grant monitoring activity performed by Cohn-Resnick in support of your grant compliance function. And those reports and their results are communicated for several pages extending through page 116. All of that internal audit and/or compliance audit activity that's communicated in this report has been verified with management and myself as internal audit to include in this annual comprehensive report of all audit activity, and I'm comfortable that it is an accurate reflection for CPRIT. The last item to point out in this annual internal audit report is on page 13-118. And that is the communication to the public as to what our fiscal 2018 internal audit plan will be. As you may recall, when we performed the risk assessment in 2016, I proposed an internal audit plan for multiple years, including 2018. Annually, we have a responsibility to sit down with management and review the risks and the existing current condition of operations to confirm that the audit plan is appropriate. We did that on August 7th, and I am not proposing any changes to the previously approved internal audit plan for fiscal 2018, which is currently presented to you on page 13-4. It's reiterated in our annual report, and I'm confirming to you that the plan previously approved for fiscal 2018, I as internal auditor support the continuance of that plan, and that's what's included in this annual report. And so looking at the last page here on the annual report communicates the 5 audit areas that we plan to cover next year. That includes the second part of the grant cycle, post-award grant monitoring, grant contracting. Together we'll perform that audit. State reporting, information technology services, and communications. Those will be the 4 audits for fiscal 2018. The annual report is provided in draft for your review and acceptance. Are there any questions on the annual report that starts on page 13-100 and goes through 13-119?
Any questions? I know it was thoroughly reviewed in the audit committee. But
Yes.
thank you.
Then I'll take us back to page 13-4 for a final debrief on looking forward at the fiscal 2018 internal audit plan. The same information is reflected here, the 4 planned internal audits, and then follow-up procedures on the internal audit activity that was performed in fiscal 2017, and closing those prior findings as our practice will continue. Timing of those audits has been communicated with management, and we are in the process of scheduling these audits beginning at the earliest October 2017 start point. That concludes our remarks for open session, and I'm happy to have any additional questions on any of the details of the report.
Dr. Rice has a question.
Sorry.
No, he doesn't.
I know between us and lunch. Yeah. I wanted to say, first of all, I think it's our chair of the subcommittee is it Mr. Montgomery is the chair?
It was the last meeting because Mr. Angeli wasn't there.
Well, anyway, I wanted to just make the comment very briefly that I think there's a lot of work that goes on in that committee. This is not self-aggrandizement, but I want to give you the comfort that we really appreciate the work that you all are doing, and that I think that the committee is a high-functioning committee because we scratch it pretty hard, if you will, through that process. And so I just wanted to make that comment. And we kind of, I think, look in a lot of detail and worry about things which are anything other than perfect. I just had one comment or question, I should say,
Yes.
beyond that. And I guess I'm just saying thank you for the good work. It's an ugly process, but somebody has to do it. And,
I don't really think it's that.
I didn't mean to say it that way.
Depends on
To your
if you're the
question,
auditor
Dr.
or
Rice,
the
to your
auditor.
question.
That's just a question.
You're the standing between
Yes,
us and lunch.
page 13-108.
Okay.
Just as an example, the private companies that had a finding on these audits, I can't remember who's doing those audits and how we follow up on those. Thank you.
Yeah, so 13-108 is a representation of the work
Sorry, just pick one of the private companies that had one of them had 5 findings identified and remediation testing will happen in February in 18 or something. Who does that? I can't remember who does the audits on these.
Yes, the grant compliance monitoring function. So that would be Vince, our Chief Compliance Officer, and his team, which currently
So it's an
includes
internal-
an outsourced partner. And so the outsourced partner that's listed on page 105 at the top here, Cone Resnick. Yeah, yeah.
Thank you.
Is that it, Dr. Rice? All right. Ms. Martin, thank you very much. At this time, the Oversight Committee will move into closed session to take up Agenda Item 16. Given the sensitive nature of the discussion about information security and potential vulnerabilities, the Texas Open Meetings Act authorizes state governing bodies to meet in closed session to discuss a security audit. We go into closed session pursuant to Texas Open Meetings Act, Section 551.076, to discuss a security audit. The following CPRIT staff shall join the OC in the closed session: Alyssa Martin, Dan Graves, Heidi McConnell, Terry Simeon, Kristen Doyle, and Wayne Roberts. The time is 1:15. We expect to reconvene at 1:45. Thank you. It is 2:11, and the CPRIT Oversight Committee is called back into session. And we're going to go, Council, could you help me with where we are on our list? Superintendent?
Page 23.
Okay. The time is 2:12. It's my understanding that the... Okay. And then here we are. My understanding, the Audit Subcommittee met on August 7th and recommends that the Oversight Committee approve the audits and annual report. Is there a motion to approve the internal audit reports for pre-award? I've got a motion and a second. All in favor say aye. Aye. Any opposed? There are none. Motion carries. Is there a motion to approve the FY 2017 internal audit annual report? So moved. The motion, have a second? All in favor vote aye. Aye. Any opposed? Hearing none, the motion carries. Okay, jump back to Dr. Garcia. The chair recognizes, it's tab 6. The chair recognizes Dr. Garcia to provide the Chief Prevention and Communication Officer's report.
Yes.
Thank you.
And in the interest of time, I think I will jump directly to the award recommendations. If you'll look in your grant book, starting on page 49. So you have a slate of 8 projects being recommended for a little over $14 million in 4 different slates. The Prevention Review Council recommended, and the PIC accepted their recommendation to reduce all of the award amounts by 10%, so that we could have sufficient funds to support all of the projects they wanted to fund this cycle. So we do have 4 slates. I've been encouraged not to go into description of those projects, but to answer questions. We have 2 colorectal cancer prevention coalition awards, 1 that was a deferred application for a continuation expansion award, 3 evidence-based services awards, and 2 tobacco control and lung cancer screening awards. You have the descriptions. I'm happy to highlight any of those or discuss any of them if you would like.
Our chair of the Prevention Committee is here. Do you have any comments?
We reviewed these in depth and had a unanimous recommendation to go forth.
Thank you. Any other questions for, observations or questions for Dr. Garcia? All right, hearing none, I remind you that Mr. Burgess has certified compliance of the prevention award process. Mr. Angeli has reported a conflict with the application PP 170082 submitted by the University of Texas at Austin. Are there any conflict of interest declarations that have not already been registered with the committee? If none, Members, you have the applications and grant amounts recommended by the PIC for the prevention grant awards. PIC's recommendations will be approved if 2/3 of those present vote to approve. Hearing none, we'll take up the University of Texas at Austin recommendations first. I'll entertain a motion to approve the PIC's recommendation for PP 170082.
So moved.
Got a motion and a second? All in favor vote aye.
Aye.
Any opposed? Hearing approval from at least 2/3, the motion carries. I note for the record that Mr. Angeli did not vote on this recommendation. I'll now entertain a motion to approve the PIC's recommendations for the remaining 7 prevention awards. You have a motion? You have a second? Didn't hear one.
Second.
Second, all in favor vote aye.
Aye.
Any opposed? Hearing approval of at least 2/3, members present and able to vote, the motion carries. I'll entertain a motion delegating contract negotiation authority to the CEO and CPRIT staff and authorize the CEO to sign the contracts on behalf of CPRIT.
So moved.
Got a motion and a second. All in favor vote aye. Aye. Any opposed? Motion carries. Now recognize Dr. Garcia to present the prevention programs plan for RFAs for 2018. Her presentation is in your agenda book at 6-3, page 6-3.
Right, so if
Thank you,
you'll
Dr.
turn
Garcia.
to the other book, tab 6-3, you have before you the Gantt chart and a description of the program RFAs. I did approach this assignment just a little bit differently, it being the first time. I hope it's providing enough information for you. But for the first cycle, which is already underway of fiscal year 18, we have 3 prevention RFAs that have already been released. They are evidence-based. This is on page 6-5. You'll see the descriptions. The evidence-based prevention services RFA, which call for comprehensive services across the continuum of prevention activities and interventions. There's a tobacco control and lung cancer screening RFA. It was a new RFA in 17-2, the second cycle of this past fiscal year. And we really wanted to encourage more applications in tobacco cessation and also support the new lung cancer CT screening for high-risk individuals. The third RFA is a dissemination of CPRIT-funded cancer control interventions, and this is really to encourage our successful grantees to package, disseminate, and to help others prepare to either apply for or implement those projects as well. In order to really speed up the process and get more of these reviewed and approved, we decided to have them be continuously open and reviewed quarterly. So they will be reviewed quarterly by the Prevention Review Council and come to you quarterly, very much like the recruitment awards are done. And this is really an attempt to just speed up that dissemination process for successful projects that we have previously funded. So that's fiscal year 18, cycle 1. For fiscal year 18, cycle 2, we had quite a bit of very good discussion with the subcommittee about a new RFA, and that one focuses on the expansion of cancer prevention services to rural and medically underserved areas. So the idea here is that grantees, applicants would be encouraged to add an additional service to a project maybe that they already have going on. So if they're providing, say, breast cancer services, they would add cervical cancer services or other kinds of services that we have on our approved list. And they would also be required to address medically underserved and rural areas of the state. So that would be a requirement, regardless of whether you're expanding to a new service or just expanding to additional counties. that requirement would be that they cover medically underserved and rural areas of the state. So that is a new RFA for the second cycle of the fiscal year. And then other mechanisms that we would consider releasing are ones we have released before, and you'll see those 3 listed. Typically what I do is I will make revisions to all of the RFAs as needed. They always need to be updated and have them ready to release, but make the decision a little bit later when I know what is coming in for fiscal year 18 cycle 1, when I know the size of that pool of applications coming in. I will have information on preliminary scores and be able to judge how strong those applications might be, and then make a decision, usually like the month before releasing, whether to release all of them or to release a more limited number of RFAs. And these, again, as I said, are pretty standard RFAs, ones that we have released before, so the community is fairly familiar with what would be contained in those RFAs. Questions about the RFAs or the schedule of release for the next 2 cycles?
I want to note the development of the county-by-county results that you now have. And when you look at this RFA, this expansion of services to rural areas, that new, more granular information is going to put us as an oversight committee and you as professional staff in a better position to really examine, again, in a more granular way, our reach into those rural areas. And I know that Mr. Margot, as chairman of the prevention committee, has emphasized the importance of that, and really appreciate the progress we're making in that regard.
Right. For the rest of the oversight committee, we have about 6 months of data right now, and it's a little bit early to really read a whole lot into that, but where the applicants are reporting county by county what services they're providing to people in those counties. So as we move forward, we'll have more and more data to be able to do a better analysis of what's going on in those counties.
Appreciate Charles.
I want to echo the chairman's comments, and also thanks for the chart on the release of the RFAs. I know that's new, but I think it's very helpful.
Thank you.
All right. Any other questions or comments for Dr. Garcia? Craig?
I do. No, I thought about including it. I knew it was going to be a very long agenda, and I can save that for November.
And we
But
can share it with you. We can share it with you.
I do have the latest one in my book, but I'm happy to send that out to the Oversight Committee. It actually looks very strong as we stand.
It is available in our portal as well.
I'm sorry?
It's available in our portal.
The map?
Yes.
I can put it up in the portal, yes, in your...
Well, it's in the committee, the preparation, the materials for the subcommittee.
That's right. And this one
So-
has been updated, will be updated, and actually has been updated with these 8 new awards and the counties that they propose to serve.
So you'll share that with
So,
the
yes, sir,
committee.
we can provide that right after this meeting.
All righty, thank you. Any other questions or comments? All right. I will entertain a motion approving the prevention programs plan for active and proposed RFAs for FY 2018, noting that Dr. Wilson did 2018, 2019, and Dr. Garcia's is 2018. Can I hear a motion, please? Have a motion and do I have a second? Have a second. All in favor, vote aye.
Aye.
Any opposed? Hearing none, the motion carries. Thank you. Dr. Garcia, now could you please give us an update on the communications program?
That's right. I was about to leave. You have that memo under tab 6, page 7. And again, I will just update you on the conference activities and let you know that registration opened at the end of July for the conference. We have 76 registrants, which is a little slow to start, but we will pick up steam, I think, very soon as people are back to school and paying attention. And we have 204 abstracts that have been submitted. So plans are well underway for the conference. We're working right now with the speakers to finalize their topics, their titles, and descriptions so that we can really hit promotion hard with that information. And you also, the other new product is the CPRIT e-news that you've been receiving daily, and I hope you like that format.
Great product.
Thank you.
Yes, it certainly is. It's really terrific. Much.
Well, thank you, Dr. Garcia. Any other questions or comments? All right, appreciate your report and wonderful progress. Thank you. Now I'd like to recognize Mr. Lang to provide the Product Development Research Program update and to present the Product Development Research Award recommendations that have been approved by the PIC. Mr. Lang.
Thank you, Mr. Guerin. Cycle 17-2, award cycle 17-2 is concluded. We had 20 applicants, one of which went through the entire process, was approved by the peer reviewers, the Product Development Review Committee, and the PIC. I'll be presenting that award momentarily. Cycle 18-1 is underway. The application process, a portion of it is concluded. We've received 19 applicants. And the review process will be starting. So those are just the highlights of my program update. I'll stop here and ask if there's any questions.
Any questions from Mr. Lang? None? All right, proceed.
Okay. So as I noted, we have one award to recommend from Cycle 17-2. The company's called Oversight. They have a novel therapy for a situation called BK hemorrhagic cystitis. It's not a therapy for cancer, but rather a therapy for a common sequelae of cancer. Bone marrow transplants, stem cell transplants are a very common procedure for patients who have blood cancers. Unfortunately, while they work very well and they're very common, they unfortunately can reactivate latent viruses that are in all of our systems, and these latent virus activations can cause secondary infections. In fact, these infections occur in about 70% of patients. So at a very high level, this company is and there's no therapy for these for the most common one. This company, Oversight, has developed a very novel technology to address this unmet clinical need. When these latent viruses are activated, this hemorrhagic cystitis occurs. It's a life-threatening situation, results in severe bleeding, renal shutdown, et cetera. So it's a very, very serious situation with no available therapy. This company's developed a novel immunotherapy for this disease. It's been clinically tested. They've conducted 2 clinical studies already with very successful results. They have a $14 million program to conduct a phase 2 clinical study, of which they're requesting $9 million of CPRIT support. Obviously, the rest would come from matching funds. So this would fund a phase 2 study. They have 140 patients to advance it into the clinic and position the therapy for FDA approval. This, again, was reviewed and approved by, as I noted, the peer reviewers, the Product Development Review Committee, the PIC, and the Product Development Subcommittee. They received one of the highest scores during my tenure here. They were scored a 1.8, which is one of the highest that's ever been received for any product development applicant. So again, there's an extensive report provided. I'll stop here and ask if there's any questions.
Dr. Rosenfeld, as chair of that subcommittee, would you like to?
This proposal was reviewed and extensively discussed at the Program Development Committee and recommended for approval.
Thank you. Any other questions for Mr. Lang on this recommendation? All right. I will remind all of us that Mr. Burgess has certified compliance of the Product Development Award process, and no Oversight Committee members have reported a conflict of interest with the application. Is there any conflict that has gone unreported that you'd like to bring forward now? All right, hearing none. Members, you have the application and grant amount recommended by the PIC for the Product Development Research Grant Award. The PIC's recommendations will be approved if 2/3 of us vote to approve it. I'll entertain a motion to approve the PIC's recommendation, an award to Oversight. Got a motion and a second. All in favor?
Aye.
Any opposed? Hearing none, the motion's approved. I will now entertain a motion delegating contract negotiating authority to our CEO and the staff and authorize the CEO to sign the contract on behalf of CPRIT.
So moved.
Got a motion and a second. All in favor, vote aye.
Aye.
Any opposed? Hearing none, motion carries. Okay. Mr. Roberts has notified the board by letter provided to the Oversight Committee on August 10th indicating that he seeks authority to disperse grant funds in advance to the company we have just approved for an award. Are there any questions for Mr. Roberts regarding this request? All right, if no questions, pursuant to the General Appropriations Act, Article 9, Section 4.03A, do I have a motion to authorize CPRIT to disperse grant funds via advance payments to Oversight upon execution of the award contract and the successful completion of tranches? Got a motion and a second. All in favor?
Aye.
Any opposed? Hearing none, the motion is approved. Okay. Thank you, Mr. Lang. I appreciate it.
Thank you.
Now, ask the members to please go to item 21 and ask Ms. McConnell to please come forward and present this agenda item, and she will present the staff recommendation for 3 service contracts. Thank you.
Good afternoon, members. In the memo, it summarizes our recommendation to approve contract renewals with the Perriman Group for economic assessment services, Weaver and Tidwell for internal audit services, and Hahn Public Communications for strategic communication services. I did send an email to the members of the Oversight Committee yesterday requesting confirmation that there are no financial conflicts. So thank you to those of you who have who responded already, and if you haven't seen that email, we look forward to your response. Happy to answer any questions.
Any questions for Ms. McConnell? All right, members, there's a motion. Is there a motion to approve contracts with the Perriman Group, Weaver and Tidwell, and Hahn Public Communications?
So moved.
Got a motion? Do we have a second? I think I heard one. All in favor, please vote aye.
Aye.
Any opposed? Hearing none, the motion carries. Now I'd like to ask you to turn to item number 19, the Myrna shareholder vote. At this time, the chair recognizes Ms. Doyle to present the recommendation regarding the Myrna shareholder vote. Ms. Doyle.
Members, you have my memo that is behind tab 16 in your meeting book. We are asking today is that you vote to authorize the Chief Executive Officer to be able to vote CPRIT's ownership shares in Myrna at the annual shareholder meeting on August 24th. The vote will be to merge with SynLogic. The board of directors for both of the companies, both Myrna and SynLogic, both recommend the merger, which was announced in May. I'm happy to answer any questions on my memo or on the delegation of authority you will be giving to Mr. Roberts.
Could I ask Mr. Montgomery to also speak briefly on this subject? He and Mr. Holmes worked with Mr. Roberts and Ms. Doyle on this extensively and very successfully.
Well, I don't have a lot to say. We had a contract provision that we thought was very much in our favor. Myrna obviously disagreed with us. They needed to get rid of CPRIT in order to move forward. And while Mr. Guerin's kind to give me any credit at all, Mr. Holmes really led the charge and did a superb job. And we were able to, can I say what we?
Yes,
Yeah.
it's
So
publicly reported now.
We were able to get Myrna to pay us $5 million in order to get out of the one contract provision. So we considered it quite successful, and now it allows us to go forward. Myrna was shutting down operations, and now we will own some small amount of stock in the new company. I don't think it's much, but
About 2%.
something.
2% of the company.
Just for the question, does that money return to CPRIT, or does that go to the general fund of the state of Texas? I had to ask.
So because the $5 million payment was a contract payment, and so that returns to CPRIT to continue to grant out in awards rather than to the general fund.
Terrific work on part of all four of them. It was really outstanding result and well above what we way above what we originally offered, right? So, great job.
Is there any problem with CPRIT as a Texas organization owning stock in a company in Cambridge, Massachusetts?
No.
Answered.
The state does, in many cases.
What number is the tab? Okay. If there are no other questions, is there a motion to delegate authority to CPRIT's Chief Executive Officer to take all actions necessary to vote CPRIT shares in Myrna at the company's upcoming annual shareholder meeting? So moved. Got a motion? Do we have a second? Have a second. All in favor, vote aye. Aye. Any opposed? Motion carries.
Will Mr. Roberts be traveling to Menlo Park to exercise the vote?
My guess is not.
Now, I'd like to ask you to turn to tab 14, please. It's amendments, pardon me, item 17, tab 14, amendments to the statutory chapters. Chapter now recognizes Ms. Eckel to discuss the proposed administrative rule changes.
Thank you. Behind tab 14, you'll first see the final order for the rule that we proposed back at the May meeting, eliminating the 5th quarter reporting period for grantees. We didn't receive any comments. Following that, you'll see the preambles for 2 new proposed rules to start the rulemaking process over again. One that clarifies that grantees need to maintain grant records for a period of 3 years, and one that prohibits grantee payments to a subcontractor that employs a relative of the grantee unless they have an exception.
Okay. Any questions for Ms. Eckel? All right, members, is there a motion to approve the final order adopting rule changes to the Texas Administrative Code, Chapter 703? A motion and a second. All in favor, vote aye. Aye. Any opposed? Hearing none, the motion carries. Members, is there a motion to approve the publication of the proposed changes to Texas Administrative Codes, Chapter 703 in the Texas Register? And second, I have a motion and a second. All in favor, vote aye. Aye. Any opposed? Hearing none, the motion carries. Okay. Now we go to item 22, subcommittee business. Moving on to the next agenda item, the committee assignments for 2018-29 recommended to the board are at tab 19. I offer these recommendations from the Nominations and Governance Committee to you for your consideration. Are there any questions for staff or the Nomination Subcommittee? Okay.
Can we institute a term limit for the service on the Audit Committee?
Yes, you certainly can.
Only based on good behavior. So you're stuck there for a while. So that's right. Okay, I believe, I'm hopeful that every member of the OC was able to secure the position that meets their interests, and I believe based on the input we got, we were able to accomplish that. Any questions? All right, members, is there a motion to approve the new subcommittee assignments for FY 2018-2019?
So moved.
Have a motion?
Second.
And a second. All in favor, vote aye.
Aye.
Any opposed? Hearing none, the motion carries. Okay. I'd like to turn to item number 23 in your... It's tab 20, item 23. The next agenda item is officer elections. Because Mr. Holmes, the chair of nominations, is not here, I will present the recommended slate of officers. The Nominations Committee unanimously recommends the following slate of officers: Will Montgomery as presiding officer, Dee Margot as Assistant Presiding Officer, and Amy Mitchell as Secretary. The Nominations Committee enthusiastically endorses these nominations and deeply appreciates the commitment of all of these members of the Oversight Committee in undertaking these responsibilities. Is there a motion to approve the slate of officers? Got a motion and a second? All in favor, vote aye.
Aye.
Any opposed? Hearing none, the motion carries. And congratulations to Mr. Montgomery, to Mr. Margot, and again to Ms. Mitchell for her continuing to serve the Oversight Committee, losing my voice, as our secretary. Congratulations to you all, and thank you.
Thank you. And as the first thing I want to do is make sure everybody got a copy of the sayings of Chairman Will. Okay? If not, I can get you some. But it is my...
The little red book?
Yeah, exactly, exactly. It's my great pleasure and honor to say that I follow two outstanding members as chairs of this board who have worked tirelessly. And in particular, since I worked as vice chair with Mr. Guerin, I want to tell everyone that the citizens of Texas can be assured that for the last 2 years, a steady hand informed by wisdom, intelligence, and the desire to help those in need has guided this agency. On a personal level, I am very proud and honored to have served on this board with everyone, but particularly Mr. Guerin. And now, I have a resolution to read, as I can read. The Oversight Committee of the Cancer Prevention and Research Institute of Texas, also known as CPRIT, by unanimous vote of its members, adopts the following resolution. Whereas the Honorable Preston Pete Guerin has served as the presiding officer of the Oversight Committee from August 19th, 2015 to August 16th, 2017. Whereas Mr. Guerin assumed the role as presiding officer during an important period when maintaining the confidence of the legislature and the citizens of Texas was critical, as CPRIT fully re-engaged in its role in the fight against cancer. And whereas Mr. Guerin was pivotal in maintaining that confidence, and whereas Mr. Guerin worked selflessly for and on behalf of CPRIT, giving his time, prestige, and energy without remuneration, and whereas Mr. Guerin steadfastly performed the duties of his office with equanimity, persistence, and poise, and whereas Mr. Guerin earned the respect and admiration of CPRIT staff through his dedication in furthering the mission of CPRIT, and whereas Mr. Guerin likewise earned the respect of his fellow Oversight Committee members through his wise counsel and able leadership, and whereas CPRIT's mission of promoting prevention, academic research, and product development advanced under Mr. Guerin's guidance and the benefit of all Texans, now therefore be it resolved that the Oversight Committee of the Cancer Prevention and Research Institute of Texas hereby recognizes the Honorable Preston Pete Guerin for his distinguished service to the citizens of the state of Texas and expresses its gratitude for his many and lasting contributions to the Cancer Prevention and Research Institute of Texas. Further resolved that an official copy of this resolution be prepared for Mr. Guerin as an expression of high regard by the Oversight Committee members and CPRIT staff. Signed the 16th day of August, 2017. Congratulations.
Thank you, Will. Appreciate it. Will, thank you, and it's been a privilege to serve as the Oversight Committee presiding officer, chair, whatever the proper term is. It's been a good 2 years, and it follows a good 2 years. And those 4 years follow a good 2 years, notwithstanding the press accounts to the contrary. And I believe I speak for everyone on the Oversight Committee. We know where the heavy lifting takes place and who does the heavy lifting. And those are the men and women sitting out there and then sitting to my right. So thank all of you for the outstanding work you've done and through some very difficult times. CPRIT's in a great shape thanks to you. And we look forward to continuing to work with you and work for you in service. But I'm honored by this resolution. I appreciate it. And it's been a high privilege and honor to work with all of y'all in this organization. Thanks a lot.
Chair recognizes Wayne Roberts.
On behalf of the staff, this one is relatively easy or relatively painless because with this recognition, it doesn't mean that you ride off into the sunset. We still have your service for a couple of more years, but on behalf of the staff, I'd like to say thank you for your service. You have been an absolute delight to work with on days.
Well, thank you.
Other days have been a little bit different.
I appreciate that, as it should be.
As it should be.
Aside from, oh, excuse me, go ahead.
On behalf of the staff, Chief,
Well, thank you. Appreciate it. Thank you all. This is-
The happiest duty one has serving on this board is to help the people who are suffering from the burden of cancer. The second happiest burden is to call for a motion to adjourn.
No.
Oh, sorry, never mind.
Work is
Oh,
not
you guys
done.
thought you could leave.
What?
Continue serving until the end of the meeting.
Okay, pardon me. Give me that back.
Or you can move over here.
That's right.
We got a lot of
Okay.
action to move.
The council tells me to keep going.
Already messed up.
Chair recognizes Mr. Roberts to present the Health and Safety Code, Section 102.1062 waivers.
Members, these waivers have been explained in the meeting book. They are essentially identical to those that have been presented previously. I would like to point out, though, that the waivers are specifically for the situations described and may be revoked by the Oversight Committee at the Oversight Committee's will. I request approval.
Is there a motion to approve the 5 proposed Health and Safety Code, Section 102.1062 waivers? We have a motion. We have a second. All in favor, vote aye.
Aye.
Any opposed? Hearing none, motion carries. I'd like to ask you to turn to item 12, Scientific Research and Prevention Program Committee appointments. The chair recognizes Mr. Roberts to present the appointments to the Scientific Research and Prevention Program Review Panels.
Yes, members, the 4 recommendations were reviewed by the Nominations Committee and are recommended for approval. I request your approval of my appointments to the Scientific
Is there a
Research
motion to
and
approve the
Prevention.
scientific? Got a motion and a second? All in favor, vote aye. Any opposed? Hearing none, the motion carries. I'd like to ask you to turn your attention to item 12, the honoraria policy. Mr. Roberts.
Again,
Thank you, Mayor Margot. Thank you. Safe travels.
Again, members, this is exactly the same honorary policy that is in effect right now. I request approval.
Got a motion, and do I hear a second?
Second.
All in favor, vote aye.
Aye.
Any opposed? Hearing none, the motion carries. All right, we've got to, please turn to item 15. I believe Paul Ballard is here, was here.
Yes,
Uh,
that's right there.
Oh, there he is, okay. Chair recognizes Ms. Doyle to present the resolution transferring management authority of certain equity assets to the Texas Treasury Safekeeping Trust Company. While she is preparing her remarks, Paul Ballard, the CEO and the Chief Investment Officer for the trust company, is here. I ask you to tell us a little about the trust company and the organization to whom we're entrusting our valued assets.
Yes, sir.
Thank you, Mr. Ballard.
The trust company is a investment management subsidiary of the State Controller's Office. We oversee about $50 billion of investment assets ranging from Treasury Pool, which is its operating funds. It's a liquid AAA rated daily liquidity. Oops, sorry, is that working? Daily liquidity fund. We have an enhanced version of that that puts up about double the return of the Treasury Pool, but it's still fairly liquid. We manage the State Water Implementation Fund. It's a $2 billion long-term drawdown fund and about $4 billion in endowments. In 2015, we were given responsibility for oversight of the former Emerging Technology Fund of the state of Texas. It was comprised of 98 early-stage venture investments, making it possibly the largest early-stage venture fund on the planet, I think. And we've been working with that for the last couple of years, and so our thought was that we could leverage that experience to the benefit of CPRIT when you all come into the possession of equity. 2/3 of the Emerging Technology Fund is life sciences, so we have, I believe, 6 cancer-related companies and a number of medical devices, consumer products, kind of across the board.
Any questions for Mr. Ballard? I know the Oversight Committee and staff have considered this thoroughly, discussed it thoroughly, and we believe it's a great fit. and it's a resource that you provide to us that we don't have, and it's great that it is in existence, so the assets that are being created by CPRIT Investments do have a place to go and be professionally managed and looked after for years or decades to come. So thank you for this opportunity to, in effect, partner with you.
Yes, sir, we look forward to it.
Thank you. Any other questions for Ms. Doyle or Mr. Ballard? Ms. Doyle, do you want to add anything? I think it's swinging in our favor. I think we got the votes, but you can.
The one thing that I would add is rather than just acting by a motion, you will be approving a resolution, and that's at page 12.5 in your book.
Okay. Is there a motion to approve the resolution transferring management authority to the Texas Treasury Safekeeping Trust Company for the assets named in the resolution? Got a motion and a second? All in favor, vote aye.
Aye.
Any opposed? Hearing none, the motion carries. Mr. Ballard, thank you, and we're very confident in placing these valued assets owned by the people of Texas with you. Thank you.
Thank you, sir. We'll try to prove that your confidence is well placed.
Thank you.
This is the last action item.
Okay, this is the last action for today. Item number 18, amendments to the Oversight Committee bylaws. Chair now recognizes Ms. Doyle to discuss the proposed changes to the OC Committee bylaws and code of conduct.
So members, my memo is behind tab 15 in the booklet. The Board Governance Subcommittee annually reviews the internal policies of the board and suggests changes. This year, they focused on new member board training, knowing that we will get at least one new member. And so the bylaws changes talk about that training specifically, that a new member may participate in an oversight committee meeting before training is completed, with the exception that they cannot vote on awards until they've completed the conflict of interest training that we'll have as part of onboarding. We also specified a time period, 30 business days, for the Oversight Committee members to complete their new member training. So the changes that you see in the bylaws and in the Code of Conduct reflect those decisions of the Board Governance Subcommittee.
Any questions for Ms. Doyle? Sound like a very good way ahead, anticipating a new member. Members, is there a motion to approve the proposed changes to the Oversight Committee bylaws and Code of Conduct? Got a motion and a second? All in favor, vote aye.
Aye.
Any audit subcommittee? Hearing none. You're going to tell me something else to do? Are you leaning this way? Okay. I'd be glad to entertain a motion to adjourn.
No, well, so we have two.
I told you. Okay. They're trying to embarrass me here in my last hour. Golly.
Can we cut the lawyers out of this?
That's
So
right.
we have two non-action items that we had not addressed: the program priorities and the chief operating officer report. We can push the program priorities impact to.
Or I can do them real fast.
You know, I would welcome your doing it real fast, but I'd like to ask that it be revisited perhaps at the November meetings, so there was an opportunity to discuss it, because this is a reflection back on the last 2 years. I think it's a very important part of what we do is to not only look forward, but to look back and be informed by that going forward. So if you would hit the highlights, and if I could impose upon my successor to add this to the agenda in November.
And actually,
Taken under advisement.
why don't we just do that? Because this is, regardless of what I say today, this is going to get reviewed at the subcommittees, and we'll be making sure that this gets the due attention that it deserves.
I think that would be more appropriate. I thank you, Mr. Roberts. Okay, what's the last one? Okay. Ms. McConnell, Chief Operating Officer's report.
I'll make this short and sweet. You have my report in front of you, have the opportunity to review it. The only thing I'll highlight is that the only thing I'll highlight is that I provided the budget, operating budget for 2018. So unless you have questions, that's my report.
Any questions of Ms. McConnell? Thank you for preparing that document, and thanks for your good work. Members, we did make a change in the upcoming meeting date in order to accommodate the demand on the staff for the conference. The next meeting date will be November 29th. I believe you've all been informed of that, but it is a change from what you had on your calendar earlier in the year. It's my understanding that the subcommittees will meet on the regularly scheduled meeting date. Okay, if there's no further business and there is no objection, I will entertain a motion to adjourn this meeting.
So moved.
Is all in favor?
Aye.
Any opposed? The meeting stands adjourned at 2:55.